So today, I hope people will be able to present their initial versions of the projects. As I've asked people before, if you could take notes, brief notes on each presentation, send those to me, and we'll aggregate them and send them around anonymously to everybody so that people can use that feedback in preparing for their final presentation. And then I've also asked people to meet in a slightly longer format to do a more in-depth review of one of the other presentations. So if there are any questions about that, please come back to me and ask for clarification if you need it. It worked pretty well last semester, so I hope it will go well this semester as well. If you don't want your presentation recorded, that's fine. Just say so and we'll stop the recording during the presentation. Boy, nothing seems to want to work today. So today I had this down for going over Elmar, Jonathan, and JH's presentation first, then Zebrafish group, then Garbiel, and then Logan and Nick at the end. If there are reasons we need to change the order, that's fine. Just let me know. And then, depending on how long the presentations take, some semesters they take all the time we have. Sometimes they don't take so long. We will continue discussing modeling network dynamics inside of CompuCell. So if we get to that, that's fine. If not, we'll do it next week. So with that, I want to go through the student project presentations. Please, again, do fill out the short comment sheets. It's perfectly okay if you want to do that by hand instead of typing it. And you could send me a cell phone shot of it, anything that we can send around to classmates so that everyone gets feedback on their presentations. Okay. Well, I'm looking forward to hearing what you have to say today. Let's start out with JH, Elmar, and Jonathan.
Hello, this is Teams. This is Trinity. We're going to present about the model about pluripotent stem cell self-organization, particularly for translation from Morpheus to CC3D. So we will recap source paper again briefly and demonstrate our app on NanoHub and explain the codes and briefly share the future plans. This is the paper we presented last time. I will call this paper as Libby et al. So, pluripotent stem cells have the potency of to differentiate into any type of cells in the body. Like here you can see that. As they divide any type of cells, they can change into the other types of cells that make up the body. They can be skin cells, nerve cells, bones, muscles, whatever cells. And there are two types of pluripotent stem cells. One is embryonic stem cells, and the other is induced pluripotent stem cells, or iPSCs. So embryonic stem cells, or ESC, are used to be used a lot for research. Still, yeah, it is used a lot for research, but it is not ideal that because you have to extract cells from the human embryo every time, either for like it is cheaper, but there is ethical issue and also many other issues. But unlike embryonic stem cells, iPSCs or induced pluripotent stem cells can be generated directly from adult cells by reprogramming them to a pluripotent state similar to that of the embryonic stem cells. Particularly these human-induced pluripotent stem cells, or HIPSCs, they are induced from human adult cells non-invasively and can differentiate into any cell type in the body, including neurons or cardiomyocytes, which cannot easily access in the living body. So this makes the human-induced pluripotent stem cells very valuable tool for studying human development, disease modeling, or drug screening. In the meantime, organoids are three-dimensional structures that you can see here in vitro, derived from the induced pluripotent stem cells or adult stem cells that spontaneously self-organize and mimic the structure and function of the particular organ. So these organoids can be used to study, again, the developmental processes, disease mechanism, and drug responses, but in a more physiological relevant environment than traditional two-dimensional cell cultures like that I showed before. So, well, what is the basic biological questions the paper addresses? Simulating organoids' multicellular behaviors to predict and control the patterns is demanded as it takes considerable amount of time and resource to create organoids in vitro. It is also necessary to test a large number of experimental conditions to reliably generate new patterns. However, this can be a very time-consuming and labor-intensive process. So this paper, Libby et al., developed a computational model to precisely control their self-organization by combining genetic engineering, computational modeling, machine learning, and mathematical pattern optimization. So our question is how to simulate organoids' multicellular behaviors to precisely predict and control their self-organization. So they first simplified the cellular behaviors of organoids to reduce complex interactions into quantifiable relationships using space, time, and protein expression. The space-time relationships were used to represent velocity of the self-organization of cells, while protein expression and time relationships were captured with a temporal knockdown over a few days. Temporal knockdown of specific molecules to manipulate their behaviors. Additionally, protein expression and space relationships were shown by characterizing spatial patterns. These characterizations can be considered minimal necessary properties to characterize and model organoids' spatiotemporal behavior. So, so particularly to manipulate the protein expression in the stem cell colonies temporally and spatially, they knock down two genes, E-cadherin CDH1 and Rho-associated containing protein kinase, or ROC1, which affects cell-cell adhesion and cortical tension respectively. So just remember CDH1 as adhesion manipulation, ROC1 will be tension manipulation molecule. So this is knockdown. So CDH1 knockdown produced distinct mosaic patterns like this due to reduced cell-cell adhesion. And ROC1 knockdown here increased membrane stiffness due to reduced cell membrane tension. This can be further manipulated by modifying the knockdown proportion of the two molecules and the timing when to introduce the knockdown. So the design parameters here are which molecule, CDH1 or ROC1, in which ratio to knock down, and where to target, and where to knock down the cell populations. And they paired in vitro and in silico models of the stem cells, and the model was able to accurately recapitulate the human-induced pluripotent stem cell spatial patterns. So, learning objectives. They adopted the cellular pots model, also known as the Glazier Grainer Hodgeweg model, to replicate or recapitulate the in vitro system in a 2D square lattice grid. The lattice on the grid, as you already all know, that represents a square region that is equivalent to 1 square micrometer. Therefore, each square on the lattice corresponds to a part of a cell's membrane or the environment surrounding the cell. To predict multicellular pattern formation, they also extended the model to optimize parameters using multicellular pattern synthesis, which is a machine learning classifier that was trained to yield the desired patterns. They adopted, for example, a supervised image classifier such as tree spatial superposition logic and swarm optimization for automated discovery of pattern producing conditions. But for our purpose in this class, our goal is to understand the cellular pots model and translate this stem cell model from Morpheus to CC3D. So Jonathan will take over from here.
Sure. So just to give a brief run through really quickly on what the app does. So you can choose between cell types. In each simulation, there's 2 cell types. One, there's just the wet type, which is the default, but you could also have cells that have CDH knockdown or ROC1 knockdown. After that, you can set the protein expression level. These are some examples here. WT at all times is at 100%. ROC1 is always set at 20%, and CDH1 you can choose. So there's a slider on the graphics display, which we can show in just a minute, that does that. And then another thing you can do is actually set the knockdown time. So as you can see, it's the time point where the specified expression level occurs. It defaults at the start, but if you want a delayed knockdown time, you can do that as well. You can also set the number of cells that are seeded. Once again, it has a default value. This time it's 50 and 50. And you can also see some plots about what's happening in the simulation alongside the graphics. The defaults with adhesion and contacts, though you can choose some others as well. So I have the app pulled up right here, and I can demonstrate it in just a second. Hold on, let me share my screen. Elmar, do you have the simulation as well? It's showing up on mine, but it's kind of smaller. I can still show it, but
I can start the simulation if you cannot show it, but you show it for you, and then you have to control.
Yeah, I'll show it in NanoHub just to give you an idea on what it looks like, and then worst comes to worst, we'll show you a larger scale version. 1 second. So here's the simulation. As you can see, it's CC3D in NanoHub. For those of you who've been using CC3D for NanoHub, this will be pretty ordinary for you. Up here we have the steering panel. This is actually a little different than most simulations that we've had for our homework assignments. We can actually change the parameters from this. And as you can see, you can choose the cell types up at the top. The wet type, CDH1 or ROC1 with an expression 20. So for the simulation, we'll start with ROC1, CDH. From here, you can choose the cell type expression. If the first one is CDH, you change it here. If the second one is CDH, You can change those parameters here. We'll start it set at 120. As mentioned before, the knockdown times are another thing that you can set. We'll start those at 0. The position will stay 50 and 50, just for this demonstration purposes. You can also show a time plot, volume surface plot, and adhesion plot. But for now, we'll just show the adhesion and contact plot. This window's kind of small, and I don't want to clog it too much. So now that the simulation is already automatically running, so if you want to get the graphics showing, you just click go right here. As you can see, we have the cell contact evolution plot, cell adhesion energy, which is also changing over time. And then tucked away here is the graphics.
You might want to try dragging the bounding box of your X11 window over to the right. You should be able to grab the bottom corner of all the way down there and pull it all the way to the right. There you go.
Okay, thanks. Sorry, I've been using CC3D locally, so I'm a little.
And so I know we didn't go through a lot of these things because most people were running local. But that can help. And then if you hit tile under window, maybe hit the tile option. Can give you a bit of a layout.
Okay, great. Thank you. So, yep, as you can see, it's running here. And if we zoom in, you can see the cells are changing their positioning over time. Some parameters of the publication that we went off of, you may remember earlier in the semester could lead to a bullseye patterning. For how it works right now, more often than not, we have multi-island patterning, and that works completely well. So over time, you can see some of these bigger clumps will split up. So something else we had, just to show you where we're at, and if you end up wanting to play around with this yourself, we have some exercises listed on this document. So it lists the cell types and all of that, and also some parameters to test. So if you have WT and WT, there won't be a pattern. For these, no pattern will form. WT and Rockland with knockdown at time 0, a pattern will form. But the best pattern will form with WT and CDH1 set at 0% with a knockdown time at time 0. Some other cell parameters to test are listed here. And I can run through a few of them, but it'll pretty much just show what this is showing right now. So yeah, and then eventually it'll just keep running just like any other simulation until you want it to stop. And if you want it to stop, change this to wait. And then you can stop the simulation.
Okay, so I want to walk you quickly through the code that we had from Morpheus and what we translated. So Morpheus had a Python file that they used to run several settings from their models to explore, and this was this Python file. And in the meanwhile, you all know a little bit Python, so I made it quicker. So the Python file, or this Python code, can be called from the command line, and you had several arguments that you can give more or less the same that we had in the drop-down menu. So you could select from CDH1 cell lines, or from this ROC20, or from the wild type. And you see as well that they had fixed values, so 0, 70, 75, and 90. And by us, we have a slider, we can set whatever we like. Then they have as well the time where the knockdown is, so this knockout time, and then the second cell line, the knockout time, the number of cells seeded. So we really took this from this file. Then they specify, so each of these different cell lines, so the wild type, the Calvin cell line, and the ROC1 cell line, they have a little bit different settings. So the size of the cell, how strong the cell is, the elasticity of the membrane, the half time that it takes, that you have half of the coding expression again. So this varies for each of the cell type. This is set here. And then the cell type that is actually chosen in between for the coding cell line. This is just a linear correlation, and they update this one by having CDH with a cell line with 0 coding expression and one with 100% coding expression, and this is just a linear correlation for all of the parameter values that they had for the cell lines. And then they use this information that they have to actually change the XML that they run. And the XML specifies the morphosis model. And then they run headless the morphosis model with this command. So that's how they do steer the runs. And then the morphosis model itself is this one. So it's everything specified in the XML, and morphosis itself have a nice user interface, but this was developed in a very, very old version. You could study it in the user interface, but we could not run it. It was just crashing. And you see that they had for every cell, okay, there were some weirdness in this model. They had the addition values. The addition values were actually negative. So they were modeling the cells like, you can think about magnets that have the same pole on the little bit floating on top of each other. And when the cells were growing, they were touching each other, and then they had contact. And otherwise, there was always a little bit media floating around. And the cells, most probably this worked in this morphosis model at the time they implemented it. But in the later version, I don't think that this still runs, and it was definitely not running when we translated this into CC3D. Then, as you can see, they measured the cells in the center, and they measured the cells on the edge of the colony. And on the edge, the cells were a little bit bigger and had a little bit different settings than they had, or parameters that they had in the center. So that's how they specified the cells, and that's quite a lot of variables. And then they had these if then else functions that they specified in this expression values. And more or less this says about if it's before the knockout time, the cell usually they don't proliferate, they just get into a state that they get ready to really proliferate and really get sorted. And if we then are after the knockout time, then the cells proliferate and try to sort. And they said that they specified a hill power function, but what they really did is they specified here a logistic function. And funny, they had initialization values, and when I read the paper, it understood the values that you give. So what they call here finals, these are really the values that you have when you have rock 1 knockdown complete, or when you have CDH knockdown. And over time, when you do the knockdown, and over time they will recover and come back to a wild type similar cell. And so you should have actually a hill curve that recovers from this knockdown type to the wild type, but that's not what they specified in their rules. Anyhow, we as well translated all of these rules, and that was the model that we in the beginning had. Then they had even a chemical field specified that hopefully they were not using it. Maybe they experimented around a bit with this one. They specified the neighborhood lattice. They specified the time, how long the model runs. Then they specified how the biology behaves. So, for example, these floating magnets, they specified with the neighborhood one. Then the cells had this velocity. this was about how the cells are seeded. This is figuring out if the cell is on the edge of the colony or if it's in the center. This is the same for the second cell line. And then this is how then the contact energy, how their contact model from cell contact was specified. Then this is the Monte Carlo step results, and here is this neighborhood order 1 that makes that the cell, even when the float medium between them, still they were quite close to each other. So, and then this described the seeding, the seeding in the beginning, the random seeding of the cell types. And this was how they tracked their data. So this was more or less what we had in the beginning, and we could run it in a Docker container, but not anymore with the available version of Morpheus. So then we went into CC3D and specified there the model, and this is just I think this, you know, right? We have 2 cell types, medium. We specified the lattice according to the values we had from them. We had center of mass, we had neighborhood tracking, and we had an external potentials, which is the force, the velocity that the cell have. We first specified just simple contact energy for the cell type, and then James helped me actually to implement the contact energy so that you have really the caterins that drive the contact from the cells. We needed to have this, and this took quite a lot to figure out what they really specified in their model was not really working with us, and we had to change this. So they had as well these negative settings that they had. So we went back to the original paper from Graner and Glazier, and we took values there. We doubled the values, and that's how we specified the media value and the values between the cell type, that we really got some sorting. Yep. So that is more or less the model. It's in the XML not that much, and PPT. The step tables looks like this. So we first had some libraries to load. We used NumPy, random, Swiss when we developed. Then I make this a little bit smaller. So, yeah. So then this is exactly the same specification. So it's a dictionary where we specify the parameters from each of the cell types. We have some constants, so Hill constants. All the lambdas were specified as constants. We made them actually a bit higher. They had quite low lambdas, so lambda 1 for the volume and lambda 1.5 for the surface. They were quite fluent cells. Then we had to calculate the MCS back to hours, and I did have some calculation, and it was about 8 MCS resulted into 1 hour. Then we had the growth step level class where we specify now the control panel that you saw in the simulation, and that's just for the control panel. And then we actually have here a seeding algorithm where we seed the cells. Is this correct? Oh no, this is where we generate the mutation CD1 cell type. We update this. So then this is the steppable, and here we do the seeding. So we read in the input parameter that were set when we have it to go, as you saw in the demonstration. Then we seed our random parameters. Then we initialize the cell type with the specified. Then we set up all the plots that can be selected. Yeah. And then the processing starts. So we update our MCS. We calculate as well the time in hours. Then we result the values that we collect for the plots, and which we go through the cells. First, we see if the neighbor, if it is an edge cell, if there's medium touching, or if the cell is inside the colony. If it's this is cell type 1, these are the settings for when it's an edge cell. These are the settings when it's a cell inside the colony. This is cell type 2. Again here, if it is an edge cell or if it is cell in the colony. What you see here, this if else, this is now what we translated from their XML. So you have in the beginning when the time is before the knockout time, and this is now the hill function that we specified. So we used the value where we could, but we really start with what the knockout time is, and we always go to the wild type. So in the end, our cells always result to be like the wild type. And this worked out fine, so the cells are now kind of sorting as good as it gets. This is just if there would be a wrong cell type. Then we update the parameters from each cell. We calculate contacts that you saw in the plots. We update the plots if necessary, if we display them, and that's more or less it. And this is the mitosis step. So when cells are dividing, time calculation, this is the time plot, and this is the mitosis division for each cell type calculated. Yeah, so that's the model. Yeah, 800 lines of code. I think that's it. So yes, so the model runs as you have seen on NanoHub, and we think we still, for the future steps, I think we can really as well now shape it a little bit, give more, make the presentation nicer when user lands on our page, and then as well do the final write-up that we have to do, and will be fast May, so it's a lot of work ahead of us, but things is running, and I think we all understand in the meanwhile CC3D and learned a lot about POTS models, and that's what we should have picked up in this class. Yeah, thank you very much.
Great, thank you very much, everyone. General questions? Gabriel's applauding. Thank you for being willing to start out. It's always hard to be the first one to go and present.
I try to somehow stop sharing. Or do I still share? Or is my
No,
microphone?
you're not sharing. You're fine.
Can you switch off my microphone? I don't find what I have to do, when I can do that.
So I had a question. You showed the simulation. Can we come back to the simulation, John?
Yeah, sure. 1 second.
If we could screen share. I guess I had a couple of questions about how things worked. So could we zoom in maybe on the cell field in that one? You could full screen that just the graphic 0 window there. Yeah. So those blue cells look like they're falling apart, and that'll happen when you have potentially when you have a negative contact energy between blue and blue. They want to create more boundary between themselves. I was wondering if that happened in the original simulation or not. In other words, I know you couldn't use exactly the same adhesion energies and the adhesion parameters, but I wondered if you went and in the original simulation, if you had wild type with wild type, did it do the same thing? If you had knockdown with knockdown, did it do the same thing?
I can maybe shim in. There are videos from the original one, and they are as well looking quite fluid. And we took the volume surface ratio from the paper. And let me quickly share again the screen, and then I can show this video. And it looks very, very, very. I have So share screen here, and I have to find the videos. So this will, I'm not prepared for that directly, but this will be fast. So now we see all how I'm organized. This is really, was it here? No, sorry, has to be here. So this is from the publication. I think this is just, oh, this is the, so you see that I think they look as well quite fluid, and you see how the shape looks from them. So
Right, but they look fairly compact. They don't look like they're striping out the way they did in the in the example in your simulation.
oh yeah, that's another thing. If you go actually to the XML from one cell type, the volume was actually commented out. If you study what they really were running. And this runs all perfectly in the Docker container, but I doubt that I think this was all running when they published, and they somehow got values, and the concept is very interesting from this paper, but when you want to translate it, this is not really going. I think you can not even translate it into a modern Morpheus version, but yeah, so that's what we had, and we really tried to stick to the value that we found in the XML as good as it gets.
But I guess I'm concerned in the CompuCell model, it looks like something is wrong with those blue cells in the simulation that Jonathan showed.
Yeah.
So that's what I'm trying to, in other words,
yeah.
I understand that the parameter values may not match, but if they say wild type and the CDH1 knockdown should sort, then you would like them to sort. If they,
yes.
And if they plot the time series of the contact areas or something like that, you'd hope that those would agree more or less.
Yes. So, I mean, when you run wild type, you'll see that it doesn't sort at all, and the sales are much more fragmented. And when you run with a setting with ROC or with CT, the cells are in the beginning much more fluid, and then when they come to a wild type, they get like stiff again. So that's what happens to this hill curve, and but it's always, the settings were always very fluid. Maybe there is somewhere an error. Everybody, it can happen that you have an error, but as far as I read the paper, I think the settings are as good as it gets to the paper, even it looks so much different.
Right. I guess I just did the one you in the specific simulation that Jonathan showed us.
Yeah.
It looked like there's something wrong with the way the blue cells are behaving.
Okay.
So I would double check that against what they predict and make sure maybe they behave strangely also in the original simulation, but that one looked a little bit odd. Giuliano, did you notice that?
Yeah.
Yeah, I think the cells are very fragmented. They don't look healthy. I don't know.
Now the blue cells are striped out, sort of zigzag, and that usually means that you have a surface area constraint in these cells.
So I have a value, and then we have the surface, this volume to surface ratio, and this is 1.12. And as you maybe remember, you said it should be at least 1.3, so or 1.1, but that are the values that they have, 1.2, 1.1, and that's why they are so fluid and spread out so much. I mean, of course, I can make them stiffer. I can set it to 1.4, but then we are not anymore doing what they did in the model at all. So what's then the point to say that we translated this Morpheus model?
Well, I think that's a very interesting question. In other words, there are two kinds of translation, one of which is trying to use the parameters they used, and one of which is trying to replicate the results that they got. And they're both worth doing. And so one question is, if you use the parameters as well as you understand them, do you get the same result? And the answer seems to be no.
No.
And the other question is, can you find parameters that replicate what they got? And I guess I'm asking a little bit more about the second one.
Yeah, for the second one, there is just not enough information in the paper and as well not in the supplementary data. So if you really dig deep and if you really try to find all this detailed information, it's not really around. So you could only get this out from the source code. So, and that's why as well I think they did a lot of work. This is a hell of lab work that they did, and all the other things that they do, the machine learning and so, and I think, in my opinion, it looked like this was years of work and they didn't achieve exactly the results they hoped for. And in the end, they just had to publish and published it. And one special thing is they always reference table 2 that has all the information, and this table in the supplementary data and the table 2 doesn't exist. So yeah, I mean, in the end, I cannot do magic. I'm a scientist and I can just overtake the values that are there, and it would much more be speculation when I now just made values that make the things look nice. I think that's not what we should do. But if you want to, we can of course polish the model that it just looks nice. But yeah.
I think it's more than, and I don't want to take too much time from everyone else. I'm not saying that you should make something that looks nice, but at some level, if you have an experiment and you're building a model, what do you want to do? You want to make sure that the cell types are the right cell types, that the areas are right, that the shapes are right, and that under certain conditions, they sort or they don't sort, the speed of sorting is right or not. And so I don't think it's inappropriate to ask the question, this is where we start. Now, what do we have to change to recover the behaviors that they got in their simulations? Giuliano, maybe I'm not explaining myself well. Maybe you can come help me say it in different words. Because I think seeing that example, that simulation, it's very impressive what you've done. It's a lot of work. I'm not trying to say that that's not great effort. I just think that maybe a little bit more tuning, you might be able to get it to work to be more convincing. Giuliano, maybe you can help me out here.
Don't fixate on what they did or use, but try to get to the biology that they actually simulated.
Yeah. Yes, I think I can think about it. I mean, it looks to be like the value that have most effect is this volume to surface ratio, and when we at least tweak this one, or we can even say that it looked in being like this and then tweak the values and say how if it improves or which value we think should be tweaked in which direction that they improve. I think we can do that. That's good. Yeah.
I mean, I think you've really gotten a lot done. And so I think in the next 2 weeks, a little bit of fine tuning, you could probably replicate almost everything that they have. It probably won't be, I mean, not the AI part, but the basic results. It may not give exactly the same parameter values, but I think you're very close to having this working. And I know how frustrating it is when things don't work the first time.
Where I can do that, where I think input would be more helpful. I have hard way still to translate their velocity value, and this is one that I think would as well have influence that the cells are moving more and through that sort more. And this is one that they had a little bit weird settings too. So they had the persistence time from 0.03 Monte Carlo steps, which is, I mean, there is only one Monte Carlo step. This is the time pace. So there would be maybe, yeah, that is one value that would need tweaking too. But yeah, I can look into it. Thank you. That's
so one thing maybe for JH and Jonathan, when you showed your code, Elmar, but maybe coming back to JH who presented the biology, you mentioned that there's cell division going on. I didn't catch that in the beginning. So in the organoid culture, JH, is there cell division happening?
Could you repeat the question?
So in the simulation, there seems to be some cell growth and division occurring. Is that something that comes from the organoid culture? Where is that coming from? You didn't mention that there was cell division going on when you do the organoid culture. Maybe that probably there's some cell division and cell death, but I didn't quite understand where that came from in the model.
I don't think I understand your question. So in the model, the cell division is going, but where in biology is based on it? Do you mean that?
Yeah, in other words, Elmar said that he did when you showed the Morpheus code, you didn't mention that there was a cell division component.
But I mentioned the self-organization, which of course includes cell division.
Okay, so that does, well, because some kinds of culture, you don't get a lot of cell division. So that's why I wanted to make sure.
Sure.
They have specified cell division, and I could filter it out, and we have to write this more out now from this XML model, but it's there. Yeah.
And you say there's an explicit force pushing the cells as well as the regular random motility.
Yes, but there is a force that is specified that they measured in an experiment, but the motion is random, so it's Brownian motion. So this is something should the motion not be a little bit directed? I mean, they have totally random motion, and there's just a force that is set to 9 in there. The lambda value, it's kind of like a lambda value that they set to 9. And I mean, I was trying out, you don't see anything much, but I was setting them to 100 when I was playing around, and then you really see that they move, and you can make them straight, and then they will like a firework, they go out that explodes. But so it works as a code implementation, but the settings that they have, you don't see much. So I could at least make the movement a bit stronger, but I think I still shoot them to be random. That's I don't know exactly. I have to go back to the paper and read about what they really try to do with this one. But they have movement that they specify.
Also, when the biology was mentioned, there was a velocity parameter, which includes the space versus time. So that's where cell division could come in, and they also show the cell division and cell migration was happening in the wet lab validation experiment.
It would be great to compare the time series plots with the experimental and simulated, the Morpheus ones too, just to see whether you're replicating. Oh, that's great. Other questions?
We carve this out a little bit more. We definitely will carve this out a little bit more.
I mean, it's already a huge amount of work. But again, this is why we wanted to have everybody present now, so that you can identify the things to clean up in the next 2 weeks. It's not that things have to be finished completely today. That's great. Other questions or comments?
Yeah, I was just curious, like, why CC3D over Morpheus? Like, that was my biggest thing that was not clear. Like, what is the advantage of using CC3D over their Morpheus modeling?
Well, I mean, we are here learning CC3D, and it was an option right in the beginning when we could choose our project. So JH and I for sure were interested in organoids, because some of our other projects are organoids. For her wet lab work, for me as well simulation work. And then this was a paper that was interesting because it really was doing modeling, and it was doing bringing it back to the wet lab. So it comes from wet lab data, goes into modeling, tries to optimize there, and then it was tested again in wet lab data. And I still think what they try to do and the approach of this paper and the methods they used, particle swarm analysis as well, and how they got the values out of the images and this rule-based thing is absolutely great. The concept from this study is really great, but I think that at one point that they haven't had the person who helped them to really specify the model that they would have needed. So I think they haven't had the person who really could help them to get the model done. Okay, but back to your question. So this was the paper. It was interesting, and we just choose it to get it into CCCD. We don't think it have an advantage to have it in CCCD, but it was something that we could learn about technique that they have used, and had an interesting paper to replicate in CCCD. Is this answering the question?
Yeah, thank you.
I mean, I'll be the first person to say Morpheus is a great package, and the model specification in Morpheus is very elegant. And so, depending on exactly what you're trying to do, some things are easier to do in Morpheus, some things are a little easier to do in CompuCell. Morpheus is a newer package. They learned a lot from what we did right and what we did wrong. We collaborate a lot with those people. Actually, next week, there's going to be a conference, a virtual conference in Seattle, and we're going to be meeting with them to talk about how to try to make the packages more interoperable, to try to make it easier to do the kinds of things that Elmar and JH and Jonathan were trying to do in their project. So actually, the Morpheus people are very interested in trying to figure out ways of bringing the two projects together. So if I were going to start from scratch, I might very well use Morpheus. It's a great package. There are some differences in terms of capabilities, but mostly they're pretty similar. Other questions or comments? Okay, I'd like to move on then, and let's hear from our Zebrafish team about what they were able to achieve. Where are they now? And I'll turn it over to you.
Okay, I'll introduce it and then I'll hand it over to Hayden, who will get us started. Our project is modeling zebrafish stripes. If you recall from our previous presentations, basically we are simulating the growth and modification of zebrafish stripes during zebrafish development. The team members are me, Carmen, Hayden, and Mike, and I'll pass it on to Hayden now.
All right, let's start off with the learning objectives. So firstly, we wish to understand how the three main pigment cells that form the zebrafish stripes grow and interact. Next, we want to understand the biology behind that stripe formation and the mathematics behind modeling the zebra stripe formation, as well as chemical field interactions. We also want to know how to test for other stripe formations by adjusting chemicals and parameters to make different mutant patterns. In our biological context here, we have 3 cell types for stripe variations in the zebrafish. They're melanophores, which are the black ones, xanthophores, which are yellow, and iridophores, which are silver. And stripe formation in these fish is based on cell-to-cell interactions between these pigment cells, and also the range between cells during the interaction. The long-range interactions occur in adjacent stripes and yellow stripes. And alignments of the stripes are aided by the iridophores. So fish growth widens the stripes as they grow, and repulsion between melanophores and xanthophores maintain black and yellow stripe separation boundaries. And as far as the simulation, the idea is to mimic the stripe formation through substance secretion. So the melanophores have a short range secretion that inhibits xanthophores and mildly inhibits other melanophores. Xanthophores have a short range signal that inhibits melanophores and mildly inhibits the production of xanthophores. Xanthophores release long-range signals that are required for melanophore survival. And that's sort of detailed in this little diagram down here.
Okay, so here we have some of the mathematics behind the simulation. So our stripe formation pattern is modeled off of a reaction diffusion model that has 3 different factors. So the lowercase u and v variables represent the density of melanophores and xanthophores, and also short range ligands produced by cells. And then our lowercase w represents a long range factor expressed by u. And then you can see here over on the left that the upper and lower limits of capital U, V, and W. So these are set to a constant to avoid unrealistic production rates. And if the equation is less than 0, it's just 0. If it's greater than our constant, it'll just be our constant. So it's like a piecewise function we have. And then our diffusion constants and global decay constants, and all of these variables we got that we put in our diffusion solver, we got from the paper, so that it's the simulation is like fairly correct. And then the negative coefficients in our equations are able to represent inhibition from the U field. So the U is dependent on negative V, and our V's are dependent on negative U. And that's pretty much what we have for the math. And then we're gonna just go over some of our background code information before showing our simulation, just so you guys understand. So over here we have just the basic dimensions, 128 by 128. That's what the paper that we based it off did. So we figured to keep it about the same. And then we also have all of our cell types. So we have a medium, secreting cells, non-secreting cells, and then we have melanophores and xanthophores as well. We also have a wall. This is generated to prevent cells from clumping near the edge. Whenever we didn't have the wall up, a lot of them tend to clump around the end. So we did this just to help prevent that. And then also melanophores 2 and xanthophores 2. These are a more mobile version of our cells. And then here's all of our contact energies. We also have a few more listed at the bottom that didn't really fit. It was a lot of contact energies, so we just figured we'd show most of the code. So for the most part, they're all set at 10. But we did increase the contact energy between the same types of cells to try and generate more cells separating. And then contact energies between the cells are greater than between the medium as well, so that they'll separate.
Yeah, so also I guess we kind of already went through a little bit of the XML, but I guess I can go over a little bit more of it. So as Carmen mentioned, there are 2 additional cell types, melanophores 2 and xanthophores 2. And these cell types we made them so that we can have chemotaxis if melanophores or xanthophores are in the wrong location. So in the paper that we had discussed, the the paper that we had, I guess, designed this project off of only discussed fields like chemical fields and secretions. So our advancement onto the project was trying to introduce cells. And one of the problems we had when we introduced cells is as the fields changed, the cell types didn't necessarily move with the field adjustments. So we had to introduce these two new modified xanthophores and melanophores that chemotax to the appropriate concentration locations. And this is actually similar to the actual biological model, because if melanophores and xanthophores are ablated if you ablate these cells and generate a field gradient that no longer supports a melanophore or a xanthophore, it will move to the right location or it will die off. So we tried to simulate that a little bit. Yeah, so as I mentioned before, basically our advancement on the project is we wanted to seed actual cells into the chemical environment, and we wanted to enable cell migration while maintaining a stable count. So that's kind of what we were doing in the stepables, which I'll go over, I guess, right now. So basically, in the stepables, what we're doing, this is just, I guess, a summary of some of the most important parts of the stepables. Basically, what we are doing is we are, can you see my mouse by any chance? Well, okay, so I guess I can show with this. So basically what we are doing here is we are seeding cells. Where's the best part to show this? Yes, so here's the best part actually to show this. Basically, we are seeding cells based on the threshold like the concentration of the U field or the V field. And as Carmen mentioned, U field is related to melanophores and the V field is related to the xanthophores. So if the concentration of that field is above a certain threshold, we will seed melanophores, and if the concentration is greater than another threshold, we will seed xanthophores. So then also importantly, what we are doing is we are checking to see if every MCS, we are checking to see if each cell is still in their appropriate threshold concentration. And if they are still in the threshold, if they are no longer in their appropriate location, we transition them to the second variation of that cell, and that cell will migrate. And if it does not migrate, this part of the code is basically just saying if it does not migrate by 100 MCS, it will die. So we are giving each improper located cell 100 MCS to transition their location, otherwise they will die. And our cell seeding is over here. We can see it's based off of, we're doing Poisson seeding. Basically, we're Poisson seeding until 95% of the total volume of the field is filled. And this will hopefully give us our separated cell shape that we are looking for, because if you just seed until there is 100% of the cell field is full, then we will no longer get like individual cells. They will all just end up clumping. The whole cell, you no longer will see, I guess, like the dotted cell pattern that we see in this image over here.
All right, so I'll just kind of explain the results of the simulation. So on this slide, we see just the chemical fields. So as we mentioned, we started with the chemical fields just to generate the stripe pattern formation. And so on the left there, you can see the 4 boxes, the 3 different chemical fields, the V, U, and W. And that's just kind of the initial startup of the simulation. And then eventually, as it runs, you can see that the pattern starts to form, and you can see those stripe formations based on those equations that we mentioned. All right, and then this is where the cells are actually feeded into the simulation. So this is kind of a lot on this slide, but if we go from the top left there across down to the bottom, it shows the simulation going over time, and you can see the stripe pattern formation developing over time, and you can also see that the cells are being seeded into the environment based on their respective regions. So at the very top, it's kind of hard to see the stripes forming until later on in the simulation, but you can see that they begin seeding in their regions, and then over time, as more cells are populated, you can start to see the stripes being formed down at the bottom left in the last 4 squares. You can kind of see those yellow and red stripes being shown in the actual cells. And yeah, so you can also see the biological how it mimics the actual biological process here, because if we take a look at the images of the young zebrafish growing into the adult, you can see it initially the kind of speckled cell pattern when it's younger, and then forming into the more defined stripes as it grows older. A couple other things, we can also see that as it's developing, kind of along the edges of the stripes as the fields are developing, it's kind of more ambiguous, and they eventually solidify into more defined regions, and that just kind of shows the developing process. Obviously, the real-time simulation happens a lot faster. I mean, it kind of is meant to show the overall developing process of a zebrafish as it ages. And then, yeah, so other things is we can see that there are other types of patterns that can be made from the chemical fields. So in the images there, we have just the straight horizontal stripes, which is shown in the simulation here that we have run. And then there's also just like the spotted discontinuous stripes. And then we can get like thicker stripes and stripes of different sizes and things like that. All right, and that will lead into the next slide, which talks about some of the exercises that we have with the simulation. And these are mostly meant to just kind of experiment with the different structures and shapes that we can get with just changing the chemical fields and seeing how this affects the stripe formation. So if we kind of go back and look at that XML, I have a little segment there to show by altering the initial regions of the secreting cells and like the shape and the size, we can start to get different patterns show up, and we can combine them with different regions to kind of get interference patterns. And so we can see the formation of these different stripe patterns. So whether they're continuous stripes, how uniform the stripes are in like size and shape, if they're curvature, you can kind of see that there's some stripes curving around the others. So yeah, we can explore the different structures that could be made. We can also adjust some of the parameters, such as the field thresholds and the strengths, the parameters affecting the cell chemotaxis interactions. And then these will just kind of show how the cells are seeded into the environment and how accurately they mimic the actual field, the chemical field representation of the cells. So how this kind of represents the actual biological process is that we can kind of explore different biologies of different zebrafish, as well as things like chemical interactions that may inhibit some cells' ability to chemotaxis to other regions, and just their ability to communicate with each other. So we can kind of explore how the resulting cell pattern looks, depending on how well they can mimic the chemical field. All right, yeah, I think that wraps it up.
Yes. So I will move on to just a quick live demo. I know we had a chance to look at the results as images, but I think it's probably best if we, I guess, actually did it live. Can we see my screen? Okay.
Yes.
Okay, so I will go through this kind of step by step. So here we are initializing a cell field. We're initializing the chemical concentrations with secretion cells, and they will just generate the initial pattern. And then, as Mike mentioned, the pattern will slowly develop over time. And then we'll notice that 1,000 MCS, that's when we'll start seeding the cells. And here we'll start seeing the cell seeding. I don't know how well you guys can see, and you will notice, so we have a couple different colors for the different cells. So these light blue ones, these light blue cells are melanophores that are in the wrong location that chemotax. And these light yellow cells are xanthophores that are in the wrong location that are chemotaxing. And we'll notice that as the cell field develops, as the chemical gradient develops, and we get the stripe patterns further and further up, we will start seeing these stripes further and further up in comparison to how they existed previously, where this whole section was just yellow. Yes, but basically, and then as Mike said, our exercises are essentially just modifications of this original starting parameter. That's the main, I guess, educational exercise is modifying our starting conditions so that we can get those dotted patterns, so that we can get those other modified random patterns, and then we see if we can actually get cell seeding in the appropriate locations to mimic actual zebrafish stripe pattern formation. Okay. You still see or we're back at the slides. Can we see the slides?
That's great.
Okay. And then just the last thing before we finish up, just some improvements that we want to do. We want to, I guess, we really want to try and make sure that our cell types, our I guess, model is as biologically accurate. So we need to work on modifying some of our constants to make our cells rounder and probably a little bit closer together too, because they do seem to be quite separate still. So we still need to play around with some of our numerical values. And we could also modify some of our thresholds to help with the distribution accuracy. And then also one more thing is we could try and introduce cell seeding earlier than just 1,000 MCS so that you can visualize the cell movement a little bit better. But that's essentially it right now. We'd appreciate any questions.
Okay, Elmar, do you have a question?
Yeah, I think it answered it, the simulation that you showed. So the cells are dividing, right? It's to seed them and then it happens through division that you get.
Oh, that's actually a great question. So the actual biological model is kind of 2.5 dimensions. So there are, below this, there is a full layer of the silver iridophores. And those silver iridophores would then be dividing into these melanophores or xanthophores. So to mimic that, we just assume there is a layer of iridophores below this dimension, and those are quote unquote reproducing or duplicating and dividing into these. But essentially what we are doing is actually just placing cells, just generating new cells that in cell field in areas where the chemical fields meet certain specifications. So we're simulating reproduction by just cell seeding, ignoring the second layer.
Thank you. And can I say, what was the cell, this name from the cells, the one that was below, also the one that you not simulate? So the xenoforce and the metaforce and the third type was on. I can't hear you anymore.
Oh, sorry about that. I was muted. Okay, yes. So the third cell type is silver iridophores. They're over here. My bad. I was muted. But yeah, this is the third cell type that we are glossing over in our simulation.
Very nice. Thank you.
Other questions? So I guess they also had an agent-based model of the same process. So in the simulation that you've got now, the cells don't change the field; they only respond to the field. Is that right?
Correct. The cell field itself is kind of simulating as if they were cells, if that makes sense. The chemical fields pretend that they are cells, and all the decay constants and the interaction between the cell fields is assuming that other cells exist, in that they are interacting as if they're cells. But when we implement that into CompuCell, we are implementing it as chemical fields. So in order to visualize actual cells in CompuCell, we are going to have to add cells of that cell type into the chemical field.
Did you look at all at the papers they had where they actually had modeled individual cells, the agent-based models that they had of this process, or you focused on entirely on this PDE model?
Yeah, we did spend most of our time focusing on this. We did want to try and shift the simulation to being entirely cell-based. It could be as easy as modifying just like the secretion origins and stuff like that. It could be much more complicated than that as well, which is probably likely the case. But that is actually a good point that we actually were discussing as a team, whether or not we thought it would be valuable to shift it to an entirely cell-based model. If we have time, I would definitely love to try that out. I will hopefully try and get the fixes in for like the improvements that I had mentioned at the end. I want to try and get that done hopefully by the end of this week. And if everything is working there, I will try and modify it to be exclusively cell-based.
I mean, what you've got already is very impressive. I think it would be interesting to try to swap out the fields and have the fields be generated by the cells instead of having them coming from outside. I mean, at some level, we know it works because they've got simulations where they've done it. As you say, making that happen in practice may not be so easy as the previous team found out. Just because somebody got it working in one methodology doesn't make it easy to do in another. But I think it's a great subject, and I think it could be very interesting to see that. Well, why don't we keep going?
As I was going through this project, I feel like it always changes a lot, right? Like it always you go through the iterations. I always start really large. Like I set my sights to the moon, and then it's just about, as the semester goes along, you just kind of chip away at it, and suddenly, you're at a very honest and different project than you initially set out to do. But so what I'm talking about, or what I'm going to be talking about a little bit here, is trying to get optimization within CC3D. So, there's a couple of different ways in which we can do that. And what do we mean by optimization? So we could mean evolution, right? That's what I specifically said that I was going to do. What we're going to be looking at is more akin to the differential programming, machine learning, deep learning side of things, because, go figure, like 100 neural network actually sometimes can run faster than evolution. So, that's just a little something that we learn out as we go. But yeah, so fundamentally what this project is trying to see how can we kind of get some sort of simple and streamlined optimization to use within CC3D. So basically being able to give the user using this on NanoHub, both an opportunity to learn a simple simulation using CC3D, while also being able to incorporate some of the things with, differential programming or deep learning. And so that way you're getting a more, a different kind of, a more nuanced kind of educational resource. So first off, a little bit of justification as to why, we would want optimization. So, here's a lot of the stereotypical cases, and this is deep learning specifically, while not my wheelhouse, is a very big rising field in AI, and same thing with machine learning. And we're seeing it becoming more and more incorporated with both how we do things in computational biology, but also just computational information sciences in general. They're more and more being embedded in the tools that we use. Like I have here, as simulations get more complex, as we choose to add more complexity, so does the data that they produce, right? And in certain cases, it's actually more wise to use a trained model instead of just brute force computing our way through a lot of these things, right? So a little bit of a justification as to why. But I'll offer a little. Oh, here also I was just saying, from an evolutionary perspective, we also can see these as reaching, typically this is talked about as fitness landscapes, and we can think about how can we best try to reach the tip of one of these hills? And there's all sorts of algorithms and whatever, but I'll share a different perspective. One that I kind of think of is that you have this parameter space, and technically parameter space, whether this is a specific task for a problem or whether this is, a simulation that you're running, this parameter space is technically infinite dimensions, right? So, it can span out. But not all of these are going to be really interesting, right? A lot of these are actually going to be quite boring and not related to what we would find in biology. So we know that, we have some subset of that parameter space that's actually going to be of biological relevance. So, and this is what we do a lot with causal or kind of mechanistic simulations, is we say, okay, what parameters are actually we most likely to see in biology? And then we explore that space. And maybe, quite possibly, another way that we can think about optimizations is more about thinking about subsets or sets within that subspace. And maybe it is that optimization can provide us with a couple of groups of agents or models or different parameter settings in which they might be of more specific interest, especially as we relate them to specific functions. So whether or not we relate them to trying to conserve, what I'm going to show here, which is trying to go to a certain position, or whatever, right? So, and why within CompuCell, right? Why not train this outside of, put it outside, or kind of merge these worlds? Well, there's not a one-to-one relation between the parameter space of what we often find in simulations, and also the way that the simulation actually runs over time, how it kind of unfolds as it's progressing, right? So one of the reasons why we want to train them within CompuCell is we actually want it to be able to use CompuCell stuff. We want it to be able to be, to both have a parameter, but also have the function of the parameter right there. So basically, I had previously talked about, and I have a couple of I went through a couple of different iterations for the simulation. I have that inchworm behavior, I have the amoeboid locomotion, but I decided that I actually want to start with an even simpler case. So what is kind of the quote unquote, and of course nothing is simple in biology, right? It's complexity all the way down. But what is a more simple base case that we could try and set up with using this deep learning, this optimization approach? And so for this simulation, we're going to do the bacteria run and tumble behavior, right? So that's what we're going to start off, and we're just going to say, oh, let's start off with something really simple. Let's simply try to say, okay, how can we get, if we know that bacteria move around the world in this kind of way, E. coli is a great example, how can we get them to find a food source without simply just using chemotaxis and the partial differential equation solvers that we use in CompuCell? So here I'll briefly share the code just for now. This is what we have for a run and tumble without a control, right? So I'll quickly stop my share and I'll switch over to that. And I'll share my desktop right over here. Sorry. All right, are you guys able to see my desktop now? All right, cool. So I already ran it a little bit. Basically, this little red dot here is a food source. And then we have a bacterium that's going to be doing this run and tumble behavior. I ran it here just so you can kind of see what it looks like when you run it for a long time. Just looks like a bunch of random squiggles, which you'd expect, but let's just restart it so that we can see this play out in real time. And I've got a scalar tracking so you can see it. It saves basically the position as a list. So the most recent position that it's at will be red and the least recent will be blue. So the food is this little red dot over here. We'll just let that play out for a little bit. And also we can see that I have a little thing here that prints basically and screams at us when it's tumbling and then when it's going to run. So right now it's tumbling, so it's just moving in this random motion. Now it's running, so it's going to make a guess. And right now there's a function in it that basically says determine. So whenever it ends tumbling, it determines its run direction. So it says it grabs the sample. So we have a field right now. So if you can see here, we have a field. It's different. It's different. So it's actually spreading out much more than it is, but it looks a little bit different. But basically, when the cell takes a sample at the end of its tumble, it takes a sample of its environment, and it just tries to say, oh, okay, where's the best position for me to go from that sample? And it takes that vector and it gives it, and it moves itself in that vector's position. So it'll give a little nudge, but actually, in practice, this isn't such a good way of doing things because it technically doesn't give the cell the best position in which to move. It actually gives us the best position to move from where it's sampling. So it's always going to be a little bit off. So, we're left in this thing where we have this bacteria, it's running around, it's not getting to where we want it to go. So what if we try to add a controller to it? So let me switch back really quickly to the slides. All right. So to do this, we decided to use TensorFlow, which is a very popular type of deep learning kind of, machine learning, whatever you want to call it, model that, you can create these in very short code snippets. You can create these massive neural networks. So what I have right here is the code snippet that creates the model that you're seeing to the right. You know, this absolutely, it's 5 input layers into 64 nodes into a hidden layer, which is into 32, and then which is into out, which is just 2 nodes in the output layer. So, you know, one of the reasons why I think that this is kind of cool from an educational perspective is that, you know, you can almost imagine like the little code snippets that we use in CompuCell. You can almost imagine something that's very similar to like this, to where you can be able to say, oh, like, you know, I'd be able to create a model for a TensorFlow snippet. You know, these are really handy for being able to kind of, you know, boilerplate code, build up from there kind of stuff. But yeah, so here I'll explain a little bit more on what it's doing. So when it's input, for the input, each of the 5 nodes is receiving 3 inputs. So it's getting the sample, it's just getting that whole sample. Then it's getting the X center of mass and the Y center of mass of the cell itself. Then it gets put into there, and the output, what we want is from each node, it's going to return an X and Y value of where the cell should go from there. So, and a part of and this is why for my own work, I'm very hesitant to use deep learning, is because, you know, there's a tendency to put a black box over this middle section and to kind of treat it as a magic black box that we can just throw any sort of input, receive the output we want, and bam, it's done. But, you know, there's something to be said about kind of going at this from 2 directions, right? So when I typically, and how I described my modeling approach to you at the beginning of the semester, is I typically start from the most minimal model possible. So the size of neural networks that I'm used to working with are actually 3 to 4 nodes. You know, so when it comes to, you know, time to work with this gigantic cluster, it can be a little overwhelming of where do you actually begin to analyze this. And, you know, for most people, this is a genuine problem of how do we understand what it is that this neural network's doing. And there's lots of really, really cool science that's going about understanding that. But of course, that's not what we're going to be talking about here. But I think that there is something valuable about approaching it from both ends. So both saying, okay, we have this causal mechanistic modeling approach. We can build these things in such a way, many of the ways that we've been doing all semester long in CC3D. But we also have this very high complexity approach in which we can we get it to do the behavior that we want? And how can both of these approaches mutually inform one another? So really quickly, what we're doing is between those 2 things, so between our input and between our output, we're just simply saying that our loss function is going to be the mean square error between those. This is how you calculate mean square error. I won't bore you to death with it, but basically we want our cell to get closer to the food, and this is the loss function that we apply to it. So now let's take a look at what the run and tumble behavior looks like with the controller. So to do that, I'll pass the screen share off to the other Gabriel Severino in the room with us today.
There you go. You can just give me the order and I can play if you want. If you want to talk about something, I don't know.
Yeah, absolutely. You can just let it play.
But I think
Yeah,
one of the things that is important to say is that here is just an example, like of the network being created inside CompuCell actually. So it's not outside. Everything is being created on the start function. That's the first thing, just because I already did one step. Now go ahead, I'll shut up.
Yeah, if actually really quickly, let's just run it really fast and then we can just show them the code really quick, just so that they can understand what's going on. So what I need to say too is that right now, this network is a newborn network. It's a little baby in the world. And it's, you know, it's random. So it's actually learning live, right? There's kind of 2 approaches that we could take to this. One of them is that I could show you a fully trained network and, you know, it just does it and wham, bam, it's done. Another that I think is a little bit more helpful education from an educational perspective would be to see the network slowly learn over time. So what's happening and what we'll see if we let the simulation run long enough is that the behavior of the cell will start to change. So it will start to be rewarded. The loss function will start to do its work, and it'll start to differ in behavior. And in theory, if we let this run long enough, we should see that it would hopefully converge. We're still working out a couple of kinks. So, a lot of this we just got working the other day, but we should definitely see that it starts to do it's just testing and learning right now. But we could just let that play for a little bit, and then I'll take you guys under the hood, quote unquote, and we can take a look at some of the code and see what it's actually doing. So again, since we're starting this without any previous training, it's going to perform. We would expect it to perform quite badly for a while. Oh, sorry, I just noticed Ibraheem said something. Computer training multiple cells at a time. Theoretically, yeah, it would probably just be a little bit more computationally expensive than what we're doing right now. We could. So actually, this is actually a good opportunity. So, Joel, would you mind bringing up the code really fast? Because it might help to actually see how we're doing this. And I'm hoping that what you'll see or what you'll find that's kind of cool is that there's not that much code to it, at least in the deep learning model. It's actually something that could be done. So right there, that what I showed you before, that function create model is how we're actually creating the network. And if you scroll a little bit down, so that's all in the start function. So then if you scroll a little bit down, we have our start. So this is all the stuff that we're doing for our run and tumble. We have the parameters, we're seeding the force, we're seeding the bacteria, we're doing stuff for plotting. Then in our step function, we have our secretor. We have the tumble behavior right there. And what happens when we end a tumble. So when we end a tumble, we grab a sample, we train the model on the sample. So, and if you can highlight where the model is being trained, we're giving it the input data that it's receiving. So the only TensorFlow code that we're showing you is the function above that says create model, and and what we're doing here, where we're actually telling the model, okay, predict from the input data, try to do the mean squared error between the input and your prediction. And this is all, so if you scroll down a little bit more, Joel, this is all to substitute for the function that we're calling determine run direction. So this is what would typically be in the code. So this is what is in the previous simulation that I showed you, where the bacteria is trying to determine the run direction. It grabs a sample. It checks where it is, it checks where the sample is, and XYZ. But so what we're actually substituting, so what we're, instead of where we would be normally calling this, we're actually saying, okay, train the model for here, blah, blah, blah. And then the only other piece of code in it is towards the bottom, where we're calling a training function that we're actually calling above. So here, let me answer I'll just take the time to answer a couple of questions. Is there a switch between learning and being, or is learning just fading out?
So maybe I can say primarily this is training function. So how are you at one point stopping to call this training function, or is this training function always called that you just
no, as long as it's running, it's continuing to train. So I mean, we can this of course can be trained or changed in the code, but what you're seeing right now is it's training while it's running, and it's going to continue to do so. But we can change that. So you can say, you know, you can say like, okay, at a certain epoch, stop training, and that's it, right?
Okay.
Yeah. Does that answer your question?
Yes, yeah. I mean, I was just wondering if you, yeah. Yes, that answers the question.
All right, cool.
Well, maybe,
Awesome.
maybe what I was gonna say is, when it trains, does it forget the previous network, or does it just update the network a little bit? In other words, does it do a little bit of back propagation in 1 or 2 epochs, or does it do enough epochs that it loses the memory of the previous state completely?
From what I know of TensorFlow's networks, so just keeping in mind that I'm not a TensorFlow expert, but I think the model that we're specifically doing here is just updating it over time.
Yeah, I can
Yes.
answer that question. So what
Yeah.
it does is every time it collects a sample, it does 100 epochs, right? It trains on 100 times of the data that it has. It doesn't forget that. But the thing is, since the cell changed the space, it's almost like it's learning a new space, right? So everything, so it's not as if it was trained in the same type of data, it's now training on a new set of data. So there is that also that dynamic happening. In theory, is not as if it was resetting the whole model. It's still learning, but it's based actually on the position that the cell is. So it's collecting its training data at the same time. So it's a little bit different. I don't even know how to assimilate that with other examples. I don't think nobody ever did something like this.
So what is the loss function? What is the loss
function? The loss function is the mean square error between the position that the cell's in and the food position.
So you know where you're trying to go.
Yeah.
Yes,
Previously
yes.
for the training, yes.
Yes, yeah, for the sake of the training, but the cell doesn't.
Okay, so how does it look after it's trained for a while?
Yeah, so in theory it should converge, but I actually don't know. So this is something that we've just recently got working. So I need to let it go. So
yeah, in that case, it would
another
be
thing
interesting
too is
to save the model.
Yep.
Yeah.
To keep running
Yeah,
later
absolutely.
for more training.
Absolutely. I just thought that it would be more interesting to show the learning happening right now rather than just show a model that's already been doing. But yes, no. Both are definitely 100%. Yeah.
Okay.
So do I understand it right? When you stop learning, when you have a trained model, then you kind of know this whole landscape that we now see. But if you would change the landscape, your cell would be powerless. It couldn't anymore fetch its food source.
Yes, that's a question for how we run the training. So typically the way that it, if we wanted this to be a really robust cell, so at the start of each simulation.
Now I lost your voice.
I think you muted, Gabe. For some reason you got
No,
muted.
you lost your mic. Gabriel,
and his speakers too.
Yep.
But yeah. But there is something odd about how you're doing the error function with it. Not exactly with it trying to go to where the food source is. That's fine. I agree with that. But it shouldn't.
Yeah, it shouldn't be the position.
The cell being two different positions shouldn't be an issue. Because I'm sure people have done something similar like this with robots that do racing in video games or robots that do platforming in video games. And they have to learn to do the platforming from very different positions in the map, which is what you're doing here, in a way. So perhaps there is another type of network or something, or style of training that would be more appropriate.
You guys hear me now?
Yeah.
Yes.
Sorry. Where did you lose me?
We were trying to understand, so the cell, so what are you controlling? Does the cell have a direction that it tries to go after a tumble, or does it, what is being controlled about the cell?
Yes, the direction is it is the direction after the tumble.
So why doesn't the cell then find the source?
Because so this simulation is showing the cell running in showing the cell learning in real time. So it needs to, it would need to train for it to actually then find the food source, or it would need to hit it from a couple of different times. It would need to learn. But what I was trying to say before I lost connection is that I just more thought that would be more interesting to show right now. But another way to run this is that you have it evaluate its performance evaluated at the end of every simulation rather than at the end of every time step, so that as you run it, both the food position is randomized and the starting position is randomized, and it gets to train in a more kind of robust way. So then it has to figure it out, and then you're having, to Giuliano's point, then you're saving the model at the end of that rather than restarting it fresh and having a new model each time. Does that make sense?
There is also the case that there is no pressure here. For example, if you see on those models, Giuliano, that you mentioned about a thing learning, an AI learning to play Super Mario or something, right? There's always the pressure of dying, right? When you train a bunch of different generations, and each one has a mutation or something different from the last one. And then you kill the ones that don't pass your threshold, and you let the other ones survive, and then you start a new training based on those that survived. And then you just do a little bit of a mutation on that. That's the next phase, I guess, for this project, because right now we just wanted to know if it would be possible to train a network at the same time that we're running a CompuCell simulation. And we're just doing, so every time you see a thing here that shows epoch is like the training, right? And we're just doing 100, as you can see here.
It's pretty fast.
Yeah, but for it to learn something, actually probably need like, I don't know, 1,000 or even more to learn a direction, It's just a test case for now.
Yeah, but 100 seems almost instant. So so the thing with death and whatever, you could penalize time to find source of food.
Yeah. See, I think what's been an interesting kind of challenge for me is coming from evolutionary algorithms. I'm used to thinking about things as a fitness function, but machine learning and deep learning actually use this is specifically a loss function, which I wasn't used to thinking about. So at least the way that I would deal with this so theoretically we should be able to change it to a fitness function where we're rewarding time spent near the food source. We could also do that. We could also like average distance between the center of mass of the bacterium and the center of mass of the food source, something like that. But yeah, definitely. But exactly to Joel's point too, is that what we would want to see is that after this is run during a lot, then we would start to see something interesting. I just figured it would be interesting to see it actually try and learn real time, but it's going to take a while.
I guess, I think you have everything set up here to do some really neat things.
Yeah. So what so Joel, I'm sorry, would you mind if I was able to share the screen?
No, for sure.
Just because I'll go back to my slides really quick, is what this is. I am awful at switching between these things live. So I think what's fun about this is that this was just a very, I mean, while difficult, it's still a very good base case for starting to think about the ways that we can implement things and all the different stuff that we could do in CompuCell in general. So, and here I also just needed to show my inchworm thing because I worked on it and I never forgot. So I just wanted to show it anyway. So that was my excuse to show that. But you know, we can also think about, you know, getting them to do a lot of different things, or you know, as a shout out to Elmar and everybody's project too, is when you start thinking about, okay, how can we, what's a kind of educational way that we can start to embed this machine learning, this deep learning within CompuCell as a framework. We wanted to see, okay, what does the base case look like for that? And we've shown the base case here. So these things are possible. It's actually, you know, it does work. And the important part is it doesn't take a million years. You know, if you want to, evolution would take, you know, forever, right? So, and, you know, I think what's also crazy is that what we've seen is you guys have seen several hundred runs of deep learning within a couple of, you know, within 10 minutes or something, right? So that's a lot. And, you know, from that perspective, and I think that's a testament to why what's cool about connecting TensorFlow and CC3D in this way is that, you know, you have this previously, this method in which to actually do it in not a lifetime, right? So kind of cool. But yeah, so many other extensions. Also, I would just say really briefly, there's also other stuff too. Joel and I were thinking about doing this with this thing called growing neural cellular automata, which is another really interesting thing from the perspective of regeneration. Here, I'll show you a really cool clip. But yeah, so I'll stop there because I don't want to chew up too much time because I think I've been going for a while now. But yeah,
I just want to say that I love this work. It's perfect, and yeah, the possibilities are very neat.
That's great. It sounded like you just said I love you, and I was going to be like, thank you, Juliana.
Oh, that too.
Sorry, Nick, I missed your question. Have you thought about collecting a large corpus data from CC and then training the model outside of CompuCell? A large corpus data from CC. I'm sorry, I don't really, off the top of my head, know what that is. Would you mind explaining a little bit?
Hey, yeah. I just meant like you were sampling as you were going, like, but you also were able to run the function beforehand and just sample. Like you could just let that run for however long, collect all those samples, save them, train, and then come back. And then that way you could try you know, like a bunch of different sizes of models or things like that.
Yeah. So what you're saying is, so if I understand correctly, let me re-say it to you, and then you can make sure that I'm understanding you correctly. Are you saying that you can that what I might do is have it run on a bunch of, so wait, have it run on a bunch of samples outside of CC3D and then bring that in and see how it performs then? Is that what you're saying? I'm sorry.
Yeah.
Yeah, okay, cool. Yeah, so you definitely could. I think from my perspective, one of the reasons why I want to run things within the space that they're actually behaving in is simply because I think that there's a lot of nuance that can sometimes be missed. And it's important for it, you know, at least like I was saying a little bit with when mapping parameter space to behavioral space, sometimes it's really, you know, those connections are important. But I definitely, I mean, it would be interesting, definitely, to see whether or not that would make a difference. For simple models like this, you know, maybe not. I don't know. Who knows? But yeah, no, that's a good question. It's one that I've thought about a lot.
I think if you collect the data before you train, you lose the thing of like seeing how the agent change after every epoch, right? After every step. You will have like a already trained, then it goes back to the thing of like having an already trained cell, right? We will just see, but now the process we just see the end result. The process of learning we cannot see, we cannot follow. if that's the case, I guess, for this, for justifying this way over the other way, I guess.
Yeah, I see what you're saying. Thanks.
So I think this is great. I think with relatively little effort, you could make it into something really dramatic, very exciting. So one thing that, I mean, partly things a lot, I mean, I'm noticing with the everyone's presentations, everyone's using it, things that we didn't come to in class until late. And so it makes it harder. Ironically, Giuliano suggested I cut out chemokinesis from my lectures. So of course, we wouldn't have gotten to it until the last week or two anyway. But I think one of the things that would be really interesting here is to go back essentially to the simulation, the persistent motion simulation we did recently. In that simulation, we don't have a run and a tumble because the tumble happens every time step. Right? We have the cell has a force on it and the direction of the force updates every time step. But you could have two parameters. To begin with, you could just have one parameter, which is what is the probability per unit time that you change direction? And the new direction is totally random. The other one is what is the probability per unit time that you change direction and how far do you change? In other words, how much do you change your angle? That's the second, that's a two parameter version. And now you have as the input, the concentration of the chemical over some history. And to begin with, it could just be 2 points, the concentration now and the concentration, say 100 time units ago. Or it could be the concentration over a period of time, like 100 times since. And now your penalty function is what's the integrated amount of chemical? It could be what's the integrated amount of chemical I've seen during that period. Or it could be what is the concentration at the end of that period. And now if you have the probability of tumbling, suppose we just you randomize the direction completely. So all we have control over is how far we go how long we go before we change direction. If that's a fixed number, how long we go is a fixed number, then you don't move up chemical gradients, you just diffuse. If the probability of changing direction goes down as the concentration goes up, then the cell will move towards higher concentrations on average. A very simple rule, which is if the concentration increased between the last time and this time, I don't tumble. If the concentration decreased, I tumble. A simple rule like that will get you to the target without giving a biased direction. You still have totally random directions. And so then you could ask the question, can the cell learn? In other words, it starts out by just having the rule is I stay with a constant probability of tumbling. Now I give it my history and I say, can I learn to do better? And I think that you would probably have to modify about 10 lines of code in your simulation to implement that. And that really would be how bacteria do what they do. And you could see, does it in fact learn the same choices that the bacteria really make in terms of, do I depend on the level of chemical? Do I depend on the gradient of the chemical? Do I depend on the recent history? Do I depend on the long-term history? And so I think the pieces, all the pieces of your code wouldn't change. It would just be details of how you change direction, what the outputs of your network are, and maybe what the inputs of the network are.
Yeah, that's I might
I might have to schedule a meeting with you probably sometime this next week.
Yeah.
Talk about that more.
I was thinking, unfortunately, I'm traveling Thursday. Maybe I should try to meet with every group for an hour on Friday. We should try to find some block, 1 hour block or hour and a half block of time on Friday where I go over projects with each group individually, because everybody's made a lot of progress. Everybody, I think, so far could benefit from a little bit more fine tuning to take really good projects and see whether you can polish them a little more for 2 weeks. But I think that's great. Again, and it's not necessary for the things you're trying to do, but to me, one of the questions I always ask when I'm building a model is the information that is made available to the cell something the cell really would have? In other words, what is it that the cell knows? What information does the cell have? And really, the only thing a bacterial cell knows is the local concentration. It doesn't know anything. The history. And the second thing is, what are the parameters that the cell or the agent could change? And in the case of chemotaxis, of course, cells really do move up or down gradients. So they're able to measure direction. But classically, bacteria are thought of as being so small that they can't measure gradients. They can only measure temporal changes. And so in that case, the only thing that they have control over is not the direction in which they move, but only the time over which they choose to or not to run or tumble. And so increasing the run length is straightforward. The idea with tumbling is that they don't lose the memory of direction immediately. The longer they tumble, the further they move from the original direction. And so in our model of persistence, essentially, you do some randomization steps where the angle randomizes over some distance. You could do that in one time step. It doesn't have to be multiple time steps. And so those two things could be chosen as a function of the history of the cell. So yeah, I'd be happy to meet with you and talk about that. Yeah, that's right. Because those are the things that the bacteria have. That's the information the bacterium has, and it's the things it can control.
Yeah, it's a decision it can make, right, with the information it gathers.
Right. Whereas knowing where the food source is not something it knows. If it knew where the food source was, it could just go there. It wouldn't have to learn. And so to me, it's interesting. As a proof of concept, what you've done is fantastic. Now that you've got the mechanism, it'd be interesting to see whether we can apply that to more realistic situations. And I think the answer will be yes. It would be a lot of fun. Logan and Nick, are you ready to speak?
Me and Logan will be talking about building a quorum sensing model, and specifically a model of a quorum sensing inhibition drug.
All right, so we're going to start off with just a quick review of our background. So for those of you who are unaware, quorum sensing is a phenomenon that bacteria use to coordinate phenotypes on population levels. For instance, like bioluminescence, if one bacteria glows, that's not really visible. That's not accomplishing its goal. So they typically will wait around until there's enough of them for that phenotype to actually make a difference. So they're not wasting energy. And these phenotypes are coordinated through a signal-dependent or a population-dependent signal accumulation in their environment. So as the cells grow and divide, their metabolism is linked to the secretion of these certain signal molecules called autoinducers. And as more and more bacteria grow in a given niche, the concentration of these autoinducers rises in a way that's directly linked to the amount of cells present. And then at certain thresholds, quorum sensing systems will begin to activate, and high cell density behaviors will begin to turn off, and low cell density behaviors will, or turn on, sorry, and low cell density behaviors will begin to turn on or off. So an example of this is like when bacteria are alone and they're floating in the ocean, they're at low cell density, so they turn on genes that prepare them to form biofilms on contact with abiotic or biotic surfaces. And they'll turn those on, they'll begin building biofilms, and as more and more of them grow, they'll build a large aggregate biofilm, and the concentration of autoinducers within this will be really high. They'll turn on the genes to then disperse from that biofilm so that they can spread to new areas. That's just one example. In human pathogens and shrimp pathogens, over 500 genes are regulated by quorum sensing. So it's something that controls a large amount of stimuli responses, as well as internals, cellular metabolism tuning. So it will also help them decide like what metabolic path to take, as well as help them digest nutrients that might not normally be digested. And we'll get into that a little bit, but they use what are called public goods to do that. So another benefit of quorum sensing is it allows them to share the energy load of producing really expensive enzymes. For instance, Vibrio bacteria settle on the shells of crab and shrimp and different types of oysters. And all of these surfaces are really heavy in a carbohydrate called chitin. And chitin is normally not digestible by bacteria, but quorum sensing Vibrio will actually, at high cell density, secrete a chitinase so that every cell in the local environment can chew up this chitin and digest it. That typically only happens after they've exhausted simpler nutrient sources, because while the process of producing chitinase is expensive, so is the process of metabolizing the products of chitinase action. Nick, if you could go to the next slide. So in this really diverse population of bacteria, we don't have a homogeneous mixture of quorum sensing cells. So we can have subpopulations pop up, and these come from wild type bacteria evolving into quorum sensing dysfunctional states. So they can be signal blind, where they just don't know what's going on. They act like an individual all the time, or they can be signal receiving competent, but production deficient. These are called cheaters. So what they're doing is they're listening for quorum, but they're not contributing to the public goods. So they're reaping the benefit of the other cell's energetic investment without taking on that cost themselves. And this means that they always maintain fitness advantages on an individual level. So if you pit a wild type cell versus a cheating cell in a test tube, the cheating cell will win. That's just as long as they constantly have nutrients, but that's just because the cheater cells are taking advantage of those wild type bacteria and using them as kind of like their workhorse to produce these really complicated products. Additionally, we don't really understand how these deviant quorum sensing organisms and how the makeup of heterologous quorum sensing populations impacts drugs, both in a quorum sensing sense, but also in a standard sense. So we don't know how cheaters impact antibiotic efficacy, or because cheaters also contribute to biofilm persistence since they're not producing biofilm disrupting proteins. We also don't understand how they impact antivirulence genes. So quorum sensing drugs fall under a category called antivirulence. They're not actually killing the cells, they're just shutting off their virulence pathways. And in the case of quorum sensing inhibitors, that's through shutting down the quorum sensing regulons that regulate virulence. And so we don't understand how these cheaters impact the overall fitness of bacterial populations given these contexts. Nick, if you go to the next one. So as far as what has been done in terms of modeling quorum sensing, there are quite a few models describing quorum sensing, but most of them focus on how quorum sensing evolved from non-quorum sensing populations. So obviously, quorum sensing didn't appear in the universal latest common ancestor. It's something that has appeared since then. And so most of the computer modeling systems focus on how cells go from non-cooperating to cooperating, or the inverse. Also why like production of public good is like spread about a quorum sensing population and how that might have come about. So the first bullet point there talks about the Melky model, and that just describes how signal production So like the AI or the autoinducers being linked to metabolism, how that is one avenue for simulating a public good. So saying that like a signal is produced based on metabolism and changing how cells process that can actually simulate pretty well how public goods are produced in a quorum sensing population. And then another model by Serzon et al. Uses stochastic mutation of like 3 alleles. So like the signal receptor, the signal production, and the signal response, and measures how heterologous quorum sensing populations will respond to different nutrient conditions as well as different structured, like, you know, if it basically is simulating like a microfluidics device, but you can imagine like an oyster shell is not a smooth surface. It has lots of nooks and crannies. And so they just wanted to get at how those different rough surfaces impact quorum sensing in its evolution. And additionally, there are some in vivo experiments that talk about how cheating impacts the evolution of quorum sensing regulated cooperation. In that figure on your right-hand side there, you'll see just some example panels. There's 12. This is an experiment looking at the emergence of cheating in a purely quorum sensing population. So they stick 100% cooperators into a test tube and let them sit for a while. And as nutrients go down, they measure the appearance of defector strains and the frequency of cooperation over time. They do this through bioluminescence because, as I kind of mentioned beforehand, bioluminescence is a common readout for quorum sensing. And they find that most of the time defectors will appear, but that also most of the time the wild type cells end up winning out over several generations, which just again supports the idea that this cooperation is something that is continuously selected for in given bacterial populations. And we're using the output of the in vivo experiments from Bucher et al. That last bullet point to kind of measure the effectiveness of our model, as well as some in vivo data I've collected in my lab here at IU.
Okay, great. So yes, we wanted to model these heterogeneous populations that Logan was talking about was talked about in a few different papers, but specifically that Zaron paper that kind of did this allelic structure, where they were using these alleles to kind of give the bacterium in a population different behaviors. So we took that. We also took from that Melke et al. Paper the idea of using the autoinducer as a regulator of metabolism, but in their paper, they just kind of used it as an analog for public good. We actually put in a public good field to our paper, but we made a few slight changes. I'll talk about that in a few slides. And yes, you'll see mostly in our results section, we were taking the results of our simulations and kind of recreating, trying to match what we were seeing to the in vivo data that Logan has collected. So as far as why to do a simulation like this, quorum sensing inhibitors are being developed as antibiotic alternatives. As Logan said, one of many quorum sensing behaviors is virulence. So it'd be great to turn this off. It also gives insights into the population dynamic side of the question when we give different amounts of different cell types. So cheaters, non-cheaters. We also have a third type called unconditional cooperators. How does the quorum sensing inhibitor impact the population dynamics? How do these population dynamics kind of work in their own, like if we just were to let them play out? So first off, for our assumptions that our model made, complex nutrient uptake. So it was part of our simulation, but this is kind of our analog for virulence, I said, but really of quorum sensing behavior. So in the model, when cells are quorum sensing, they're secreting public good. And when this public good is at a sufficient concentration, then complex nutrient can be taken up. We also have parameters to put in what's called the stringent response. This is a stress response that bacterial populations will go through. It's in the context of quorum sensing. something that mostly affects the defector cell types. So what we'll see is in populations that are really dense, when there is not a lot of food, it's going to take on a metabolic cost or kind of a stress cost that these bacterial cells are producing, like this extra response, this specific response to that, those conditions. And then lastly, the metabolic energy was kind of a big part of our model. It represents the resources stored by the cell. You need a certain metabolic energy for mitosis. You need, your growth is determined by the amount of metabolic energy you have. If you are running too low, you die. So what you'll see, not for the next slide, but what you'll see is we use metabolic energy for a few different processes. So starting with our cell types and fields, we had 3 cell types, just a wild type. They produce that public good if the autoinducer levels are above a threshold. So that's when quorum sensing is turned on, and they stop production if the autoinducer is below. So the autoinducer is secreted by every single cell. It says down here, it is essentially a measure of population density. So the wild type will start quorum sensing when population density is high enough. The unconditional cooperator is always producing the public good. Even if the population density isn't very high, it will be producing. And then the defector, our cheater, never produces the public good. It just reaps the benefits. So as far as our fields, we have the autoinducer, which I talked about already, the public good that's produced by sensor positive cells. That's the cells that are actively quorum sensing. We have 2 nutrients, normal nutrient, which is present in the initial conditions. We also kind of have a rewash function. After every 500 MCS, we add a new layer of nutrient. Complex nutrient, which is it is another nutrient, and it adds to the metabolic energy, but it's kind of how we sense how we measure quorum sensing in our population. When complex nutrient is being taken up, quorum sensing is actively happening. And then quorum sensing inhibitor, this is our drug, and that's added in by the simulation as well. So metabolic energy, I'm sure everyone was waiting to hear about this. So the cell dies if our metabolic energy is 0. It was going below 0, so we have a less than or equal to. There's also a biological phenomenon where if a cell is starving, it will have more stress on its DNA preservation, leading to more mutations. So when metabolic energy is below 500, there's a higher chance of mutating cell types. There's an upper ceiling of 2000 just to stop it from going crazy. Mitosis, we have if the metabolic energy is over 200 and our cells are of sufficient volume. And our metabolic energy is modified using this equation down here. So it's modified by total nutrient, which is our nutrient uptake, complex plus normal. We have a decay rate and a growth factor, which are both, the decay rate is a constant parameter. The growth factor is constant as well, but I'll talk about it later in the slides. And then a decay modifier, which is specific to cell type, that I'll also talk a little bit more about. But just in a nutshell, the metabolic modifiers that we have for the wild type, if they are quorum sensing, of course, those extra metabolic pathways will take on resources, so it will decrease. So you see our metabolic energy that we have now, plus the amount of nutrient subtracted by this big decay, call it a decay accumulation. For unconditional cooperator cells, they also get that active quorum sensing decrease, but they have an additional metabolic subtraction if they're quorum sensing and there isn't enough autoinducer in the environment. They take on an even bigger cost, obviously because there would be no benefit to quorum sensing. And then the defector cells, this metabolic consequences is what I was hinting at with that stringent response. If nutrient is readily available, is not. If there's not a lot of nutrient and the population density is high, there's this metabolic consequence that they're in that stringent response. Okay, the cell type decay modifier. So I talked about this a little bit, but just to get the math in there, the metabolic consequence for quorum sensing is just a constant value that I believe was equal to 0.05. The unconditional cooperator had that early quorum sensing cost. If there is not enough population density or autoinducer, there's an additional metabolic cost. So this distance to threshold is, the threshold is when population density is sufficient to not have this cost. So the distance there, if there's a little bit of cell density, we don't want as big of a cost. If there's no cell density at all, we want a pretty large cost, and that we used a log equation just so it didn't continue getting larger and larger. And then our metabolic consequence, our stringent response, is the population density represented by the autoinducer divided by the total amount of nutrient. So if there's a little bit of nutrient and large population density, there's a large metabolic decay for the defector. And just as a response, these fit right here in that, or a reminder, these fit in this red block. For the growth factor, so this is how much the cells grow. This also comes from our metabolic energy. And then of course, if a cell is starving, it will go into a state that says there's not a lot of nutrient. I also don't have that much stored up, so we need to grow even slower. So growth factor is decreased an additional 10%, or 10 factor of 10, so that the cell grows slower and thus incurs a smaller metabolic cost. It's trying to survive. Secretion, autoinducer uptaken every MCS secreted by every cell type. So this is like quite literally just an analog of cell density, but of course it's biologically realistic. The public good, so this is secreted by cells that are actively quorum sensing, uptaken by every cell, everyone gets the benefit. Nutrient, the same, taken by every cell. Now the amount of nutrient you take is modified by public good. So in quorum sensing populations, if there's a lot of quorum sensing, it's going to make picking up nutrient a bit easier. And then the complex nutrient, of course, uptaken, determined by public good uptake. Only if you have public good and not enough nutrient do you start digesting this more complex nutrient. So this is kind of like our hard shell that Logan was talking about earlier. You won't eat it if you have food, but if you have enough cells, you can produce the complex enzyme, then you want to eat the complex nutrient. So our quorum sensing inhibition drug, this is the one that Logan works with in his lab, called PTSP. It decreases the secretion of public good in our simulation. The amount it decreases it is determined by this equation. So the IC50, that stands for the inhibition concentration. So it's the concentration of the drug at which 50% of the inhibited behavior is seen, is presented. So this is the equation we used. I graphed it out and you can see the concentration we wanted was 0.35 micrometers. For our equation, 0.35 quorum sensing inhibitor leads to 0.5, cutting in half our public good secretion. And it has a ceiling at 1, of course, because you can never secrete more than 100% of your public good secretion, as this is a proportion of how we're doing it. Lastly, but not, of course, not least, mutation and chemotaxis. We want a mutation in there so that we could start looking at some of the results that we were talking about earlier with the different cell types emerging and the play between them. And of course, it's biologically realistic. It occurs after 500 MCS, random probability of switching cell type. So instead of having alleles, we just represented this with the cell type. It made it a bit easier to visualize how we were doing everything. And as I said earlier, if your metabolic energy is low, there's a higher chance of mutation. Chemotaxis, all the cells chemotax towards both nutrient and complex nutrient. Complex was favored a little bit, and then if nutrient is decreased, complex is favored even more. So yeah, I'll turn it back over to Logan for some outputs. Hey, Logan, you're muted.
Sorry, just harkening back to what we first presented when we talked about this project for the first time. The goal of our model is to have two different outputs, a quantitative and a qualitative one. So on the quantitative side, we have plots describing the population size of each allele over time. In addition, we also added a plot that shows the fraction of our cells cooperating. So kind of similar to that paper we showed you at the beginning, rather than the two cooperating cell types individually, it lumps them together with one line where anything above the line is a cheater, anything below the line is a cooperator. And that's just an easier way to visualize quorum sensing as a whole. Additionally, we have a field showing the number of active S states, so the number of cells that are actively quorum sensing at any given time. And then we have the overall virulence of a population. This is measured as the function of the product field and digestion of complex nutrient. So just to kind of expand on that a little bit, that complex nutrient, the digestion of it mimics what we would see in a virulence response, where the cells are quorum sensing, they're activating certain secretion mechanisms, which is analogous to this public good. And then there's an environmental response. In this case, that's an analog in the digestion of the complex nutrient. It was just simpler to link this to metabolism rather than simplifying a very complicated process like type 6 or type 3 secretion. It was just easier to do this through metabolism because it still produces the same result in vivo. So there was no reason to go down a computationally more expensive route. And then we also have the metabolic energy plotted for every cell at any given time. So you can kind of see the patterns of what cell types are increasing more rapidly given dilution time points, and what cells are decreasing more rapidly. This helped us a lot with parameter tuning. It was really nice to be able to see the slopes of the lines changing based on the conditions the cells were in. And then qualitatively, we have the spatial structure of allelic populations and their shifts over time. So the cell field has the 3 different colors for each of the 3 different cell types. It allows us to assess how, based on the inoculum, how the cells structure themselves in the population. And we'll get into some of that cool stuff here in a second. And then we also can look at the nutrient field structure dynamics. So as the cells chemotax and the nutrient does its limited diffusion, how is this field changing and how are the cells adapting to this change? And then we can also look at how different drug gradients influence the population structure and the overall virulence of the population. So the learning objectives for our model are to show how autoinducers accumulate in a population density dependent manner. This is a great health checkpoint for our model. If this doesn't happen, then there's something very wrong. So this was our first major thing we wanted to demonstrate with the model. And then also it's our first educational exercise where everyone can just see, if you're new to quorum sensing, this is what a quorum sensing population will look like and how they're producing their signals. And it can also show us that the autoinducer field accumulates differently under different population densities, which we can get into in a moment. We also want to show that complex nutrient concentration is inversely proportional to the concentration of public good, but that there is a delay in the switch. So as the public good concentration increases, after a short delay, the complex nutrient will start to decrease. This delay is representative of the time it takes for the genes to switch on, become expressed and active. We played around with it at first, and when we didn't incorporate this delay, it's not as biologically relevant because genes don't switch on instantaneously. B, we just had problems with the cells eating the complex nutrient really, really quickly, which isn't realistic either. And then number 3, we want to show that inhibitor molecules significantly decrease the activity of downstream quorum sensing behaviors. And we want to understand how different gradient distributions change this effect and how potentially different inhibitory concentration, 50, so IC50s, will also alter this as well. And then we want to demonstrate the phenotypic flexibility of wild-type quorum sensors and why quorum sensing is so powerful. So it's a really energetically expensive behavior to undertake, but it's persistent in a ton of populations. In fact, the primary quorum sensing signal is produced by 50% of eukaryotes, and that's just what we've looked at. Most eukaryotes haven't even been assayed for these different molecules. So 50% of known prokaryotes, I'm sorry, not eukaryotes, 50% of known prokaryotes actually produce quorum sensing signals. And then for our last learning objective, we just do a parameter walkthrough. We have like in the code itself, we have boilerplated in like explanations for every parameter. We're working on adding a steering window. And then and my job is to produce the educational materials. So I'm also working on like walking people through what each parameter does and having them do examples. And if Nick, before we move on, if you want to bebop over to the Google Doc showing an example of the educational materials. If you could scroll to the top for me.
Yep.
So this is just the autoinducer accumulation in a population density dependent manner. So I think it's really important for everyone to like understand the basics. So we walk through here, you know, what bacterial quorum sensing is, why I'm using a CompuCell model, and then I have people reflect on like how they think based on what I've told them. Density, like inoculum density, inoculums composition and shape will change this autoinducer uptake field. And then I just kind of walk through exactly how to change these parameters. And at the end, I have them do different experiments looking at how the autoinducer is. And I give them an in vivo data example for them to compare, as well as an example from the model that I ran. And then I explain why there's a U shape. And if you could scroll down a little bit more, Nick. And then I give them examples of what they should see given certain changes. And then I give them space to just like take notes on what parameters produce the results they're seeing. If you could go back to the PowerPoint, that was that as far. So that's the plan for all of them. The reason why they're not all done is because we are spending a lot of time designing and implementing these learning objectives. At the same time, we're updating the code for the NanoHub as I get more feedback from my thesis committee, and we continue to tune the model. So that process is a little bit slower, but we have like the final version of the code that we're going to put on NanoHub. So by the end of this week, I hope to have the rest of the learning objectives mapped out just like that one. If you could pop to the next slide for me, Nick. So this is a video demonstrating what I just talked about. You're going to start to see an autoinducer uptake increase as the cell population does. So that initial spike is because the cells start in a close inoculum, and that mimics like back dilution of quorum sensing cells. So when you grow quorum sensing culture, you have high cell density quorum sensing cells, you back dilute them for an assay. So you start at a peak, it dips back down in that U shape and will slowly climb back up to high cell density as the population size increases. And so what I can hope you appreciate is this difference. That initial peak is actually not as high as what the cells will reach. If I let this run forever, they do plateau. That plateau is more arbitrary than it is in real life. The cells will always produce autoinducers, so it doesn't actually plateau. But just for the sake of the simulation, there aren't periodic bounds, so it kind of reaches a maximum concentration for them. And if you want to click next for me, Nick, there should be, yep, an animation. Okay, I don't know why the video is in front of that. Yeah, there we go. So you can, I hope you can appreciate, no, I think it's still gonna.
Oh, that's crazy.
Yeah, I don't know why it's saying that. So what I hope you can appreciate about this is that these curves actually look extremely similar. Oh, can you go back to that? No, thank you. I think I think no matter what, it's gonna. Yeah. So if you look here, you can see also at this example curve, there's an initial spike that's lower than the final plateau, and there's that same U shape as the population grows. The x-axis there is OD 600. It's a measure of population density. And the y-axis is bioluminescence, so it's just our readout for quorum sensing. And I was able to mimic the curve almost exactly. The values and numbers are different because the in vivo data I'm looking at billions of cells, and this model data I'm looking at a few hundred. So the values are arbitrary. It's the shape that's important here. So I hope you can appreciate that we were able to capitulate that pretty well. And then moving on to the next one. What I'm demonstrating here is that inverse relationship between complex nutrient and public good. So you can see in the top left-hand corner, a complex nutrient field, and the top right-hand corner, the public good field. And then you'll see the quorum sensing sensor. So as things turn red in that bottom left-hand corner, that indicates quorum sensing is happening. And then you just have the normal cell field at the bottom right. So you'll see that public good is produced initially. That represents that initial puncta you see at the beginning. It'll go back down, and the colors fluctuate like crazy because the maximum value is decreasing so rapidly. So that's normal. But you'll see what I want everyone to pay attention to here is that max value for the public good in the bottom of the public good field screen. That'll be important for a comparison later. But you can see as the cells grow, more and more of them turn on, more of that public good is produced. And after a few minutes, as they start to run out of nutrient, they'll stay at their maximum metabolic energy, and then that'll start to come down, and that's a starvation signal based on our code. And that starvation signal will activate uptake of the complex nutrient, which you can see that minimum value of the complex nutrient field has begun to decrease. And you can start to see yellow appearing around the concentration or the cells where the concentration of public good is the highest first, and then bleeding outward. This is also a parallel to in vivo data. It's harder for that in vivo data to like make sense for someone outside of the field. It's in like cell counts and like biochemical assays. So I just stuck with our model here rather than showing that as well. But you can see that the cells then run out in that same pattern where the public good digestion began is where it first is depleted, and it spreads out and away from there as well. And then if we want to move to the next slide. So what I wanted to look at next is, okay, can I capitulate the invasion from rare that the first in vivo study we saw had? So when I run this, the in vivo study did 2,000 generations with several back dilutions within each generation. I capitulated one generation just for the sake of like time. These simulations have a lot of fields, so they run slow, not horribly slow, but 2,000 generations just was not feasible. So I did one. And you'll see that I start with almost all unconditional cooperator cells here. So that's a 96% cooperating population and only 4% a defector population. I started it this way because I'm not doing as long of a simulation. I didn't want mutation to be a factor here. So I'm just inoculating an incredibly small ratio of defectors. And the results are similar. Because when you do the same thing in real life, it's the same as allowing them to evolve naturally on shorter experimental timelines. But what I hope you can appreciate is that the fraction of cooperation starts really high. And as the simulation progresses, it dips down low quite quickly. So by 10,000 MCS, which is one generation equivalent to the in vivo data, there's only about 10% cooperators left. And those defector cells maintain a pretty high level of the population. You can see in the fraction of the population by cell type, there is some wild type cells appear. Mutation does happen like later in the simulation. I didn't shut it off. I just didn't rely on that to create my phenotypes. I wanted to inoculate that small proportion to see how that would work. And then if you can click next, we'll take a look at the in vivo data. So this is the data pulled from the Melky study. This is the population where they started with mostly cooperator strains and just a little defectors. And that bottom x-axis is the full 2,000. So what I hope you can appreciate is that when you look towards that beginning of the x-axis, we're talking the first few generations, it hits the 10% frequency, which is what I'm seeing as well. So that was a really good way for us to check the health. We do see the invasion from rare of the defector strain, just like the in vivo data do. And then if you want to click next. So now that we've shown all of this, we want to take a look at our inhibitor. So Nick, if you want to play the video on the left for me, this is what the quorum sensing population looks like with no active quorum sensing inhibitor. So the field is there, but we have a parameter in our code where if you set it to false, it doesn't do anything. And you can see this is a heterologous population. The green cells are unconditional cooperators. They're producing that public good in the top right at all times. And you can see the green dotted lines in the metabolic energy fields. They do worse at the beginning as the cell density is lower, and they're incurring the cost of producing goods that are unnecessary. As cell density increases, you'll start to notice that the wild type cells are also secreting the public good. So that's the green barrier around those blue cells. And you'll start to notice the complex nutrient tick down as they've exhausted the simple nutrients and are beginning to starve. What else you'll notice is that the red cells that are on the border of the blue and green populations will fare better than those that are in the center of the defector inoculum. And overall, the wild type cells will persist the best. And that's because they're able to toggle on and off these behaviors. And they're also able to properly regulate their stringent response, which the other cell types are not able to do. So what we'll see here is pretty quickly those universal cooperators die out, which matches in vivo experiments. The defectors in wild type persist, but ultimately the wild type is left. So that's really exciting. This demonstrates that there is a benefit to this quorum sensing behavior. All right, Nick, and before we close out, if you look at the public field, a good field, you'll see the max value is around 70. So that's what we would call like a normal amount for producing this quorum sensing behavior. And then Nick, if you want to go to the next video for me. So this is a constant concentration of our inhibitor. This mimics a 10 micromolar concentration, which is overkill. I just want to show that when you add it at this amount, you'll see quorum sensing inhibition. So what you'll notice is that there's still the wild type cell or the unconditional cooperative cells are still excreting public good, but that number is far smaller than it was previously. We're talking 0.3 instead of 70. So we're several orders of magnitude decreased. And the way drugs work, they are constantly associating and disassociating with their targets. So nothing is ever going to be completely shut off. So this was also really exciting. It's following that logarithm or the reciprocal function Nick and I established for the drug activity. And what I'll hope you notice is that the cells are actually chemotaxing more because the quorum sensing cells are not able to immediately start digesting their complex nutrient. They're seeking other things out. So the cell populations are more interpolated and mixed. Additionally, you'll start to notice that the cells begin dying off at a lot faster MCS. So whereas before the cells were persisting close to 2000 MCS, we're already having cells pop out of existence at 1500. So that's also great. It means that the cells aren't digesting that complex nutrient, and which you can see they are doing it at a low level, which still mimics drug activity. But where we were seeing depletion to like 0, where the minimum value here is 46. So we're not actually seeing a normal depletion like we would in a non-treated population. And you can see that because there's no gradient forming the cells, just low level quorum sensing activity, which is exactly what we expect. And then so at the end of this video, you see that the wild type cells are still the only ones left, but that they're growing, that the lines at the bottom are all forming an asymptote towards 0. They don't actually asymptote towards 0. They will eventually die off if we let that run long enough. They just persist a lot longer, even in the presence of the drug. And now what we're going to look at is a drug gradient field. So this is a field from left to right. So the highest concentration is on the left, lowest is on the right. And we're going to see how that changes things. So that left-hand concentration is 50 like before, and it goes all the way down to 0. So you can see there's still that low level of public good being produced. Like I said, that's normal. No drug is 100% effective. And that's why we set that function up as a reciprocal. So it would asymptote towards 0 because the concentrations you would need to get essentially 100% would kill the cell anyway. So what you can see now is that max public good excretion is actually a little higher than before, but that high value are coming from areas that are low in drugs. So the right-hand side of the field is where we're seeing higher excretion of public good. Now it's still very decreased. Like I said before, we were at 70. Now we're just in like 1 or 2, but before we were less than 1. So it's increased the amount of public good present, but it's not doing so to a level that mimics untreated conditions. And as we let this run, you'll notice that the cells begin popping out of existence in a similar time frame. This is the death, but we'll start to see on that right-hand side that the complex nutrient is actually being digested because enough of the public good is being excreted at that low concentration for quorum sensing behaviors to work. And what will happen is that wild type cells will begin to migrate over to the areas where the drug concentration is low because they are able to access nutrient that is otherwise unavailable to the rest of them. But once again, the wild type cells are the only cells that are persisting this late into the simulation. All right, Nick, you can pull out of that. So if you want to click to the next slide for me. So This is really cool, and it mimics what we see in vivo data. Wild type cells persist, that's great. The problem is that the claim in the field is that quorum sensing inhibitors do not convey selective pressure because, as you see, those wild type cells, they persist. It's the non-quorum sensing cells that are being selected against. And the problem is bacteria evolve around everything, and quorum sensing is a very, very energetically expensive behavior, and it's a very, very powerful behavior for the cell. But the problem is most of the experiments evaluating these drugs focus solely on phytonic behavior in nutrient-rich culture. So what I wanted to see in this simulation is what happens if I cycle the nutrients, if I allow the nutrient to get really low, and then I bump it back up, and then let it get really low, and then bump it back up again. To mimic passaging of cells, but instead of completely back diluting the cells, I'm letting the original population stay. So I'm just influxing and outfluxing nutrients constantly. And what you can see is that in both cases, wild type still wins. So this is inverse to the prediction of the predictions present in the field. Most people predict that as nutrient cycles, that the cells will actually incur a fitness cost to the drug being present and evolve around it. You can see a difference that the mutations happening in the non-inhibitor treated cell that persists are the unconditional cooperator cells, and the mutations happening in the inhibitor treated ones are defector cells. So that is one key difference, that maybe we're selecting for defector mutants over time. But in the same time frame, when wild type takes over the whole population, we don't really see a difference. The wild type cells are still dominating the population. And actually today I let this run for like a total of 30 cycles. And over time, the wild type maintains a similar fraction. Like I said, this is inverse to what we would expect. And obviously, like, this is a cell population of, 230 cells, not several billion. And I'll talk about the limitations of that here in a second. But what's really fascinating and actually exciting about this is informing my current in vivo experiments. So my undergrad mentee and I actually just today set up a competitive indice experiment where we are doing wild type and Luxar mutants or defector cells and competing them in nutrient conditions where we cycle them through high and restricted nutrients. And then, hopefully sometime in the next few weeks, we'll play out to see which cells dominate the population of both the treated and untreated groups. But this was never an experiment we had considered doing before, because the consensus in the field was that resistance will develop. There's no reason to test it, because in the tube we don't see it. So we just think that our lab conditions are affecting the quality of our experiments. But using this model, we were actually able to design experiments that really get at what we're trying to look for in terms of our hypothesis. If you want to bump to the next one, Nick. So limitations. So as I was talking about, our cell populations are really small. Bacterial cultures typically exist in the magnitude of 10 to the 9. So you're talking several billions of cells per milliliter, and I'm looking at 200 in a box that's closed. So this can suffer from something that's called Simpson's paradox. So small populations are really vulnerable to false trends relative to the larger meta population. So that's a huge limitation of us. So if you look at this graph, an example of this is the blue group, it looks like a positive trend. The red group, it looks like a positive trend. But when you examine the groups all together, there's actually a negative trend in real life. So while it's exciting that I'm seeing wild type persisting with and without drug treatment and any mutations being negatively selected for, that doesn't mean that happens on a meta population scale. And that's why we're really excited to do the in vivo experiments to confirm what we're seeing. Also, quorum sensing phenotypes are tuned by multiple different regulons. So the model is vulnerable to unknown assumption-derived pleiotropic effects. So what we don't know is what we don't know that quorum sensing regulates. So like I said earlier, we know of it's 700 genes that it's regulating, but we don't know how. This is just based on RNA-seq data that my lab produced. And we don't know how shutting quorum sensing off affects the regulons that regulate these phenotypes in parallel. So there could be some pleiotropic error. Pleiotropic meaning when you get rid of something, you have an unexpected effect due to the absence of that thing you're getting rid of. An example of it being the stringent response. We don't know very well how the stringent response responds over time to the lack of quorum sensing. We know that it's important that they exist together for the cells to continue to do it, but we don't know if defector cells will over time evolve to care less about the absence of quorum sensing than they do initially. Because you have to remember, when we make mutant strains, we're not passaging them a bunch. They get made, frozen, and they get taken from that original stock. So they're not really evolving between experiments. Additionally, the import and export of the autoinducer and quorum sensing drug is currently unknown. We don't really know how they get into and out of the cell. It's hypothesized that they use the general pathway called the OMP pathway, which is an osmolarity, very promiscuous importer system that bacteria possess, but that's never been confirmed. And that's something my lab is currently working on. In fact, it's one of my other undergrads' senior thesis project that they're working on. But yeah, we just assume general uptake and secretion. It's modeled like simple diffusion, but has a maximum based on concentration. So that's another assumption that we could find is just not very biologically relevant.
Okay. So I'll talk about future directions, and then maybe I'll pull up the model right after that. First and foremost, we definitely want to add a steering window. I'm not going to show it as a presentation just because my computer is getting a little glitchy. I'm adding a steering window. We really want this, like Logan mentioned, he's using it for his own thesis work, but we were envisioning this model being useful for all sorts of quorum sensing research. So to allow people to use the model and change different parameters, adding a steering window. Legends to the figure, just to make them more interpretable. Finished NanoHub, of course. Right now we have an old version of code that's up and working. We need to update it, but we have put some documentation and started getting our information up and just preparing for that. Another thing we talked about adding, I'm not sure if this will make our NanoHub, but it's definitely like something that we're talking about doing if we have time, is different bacterial populations can be different sizes and shapes. I know certain kinds can be elongated, so having that would allow more flexibility. The same with changing the drug parameters, just giving more flexibility into you're a biological researcher, you want to simulate an experiment, let's make it as close to your biological reality as you can. So can you guys see my CompuCell screen if I pull this up? No, you can't. Let me reshare. Okay. How about now? Does that look a little better? Yeah. Okay. Way. So let's see what our different graphics are, or if you're going to run for me at all. Okay. Interesting. Not sure what's going on down there. You know what? Let me relaunch CompuCell. I know that it has problems when I zoom, but you'll just have to deal with it today.
Have you updated to the latest CompuCell release, or you?
Most likely not, but I'm not sure.
It's supposed to fix some of the graphics bugs.
Okay.
No promises though.
Quorum sensing uptake. I'm going to delete these windows that aren't doing anything, and I'm going to add a new graphics window. So just so we can see here, here's our cell population. First of all, here's just our quorum sensing inhibitor drug. So in this particular configuration, it is even all the way through. Metabolic energy, so they're doing the best on the outside where there's less cell density, less uptaking nutrient. And just to show why, we can show that nutrient is obviously depleted most in the middle, complex nutrient. So just to show you that we really made those plots from our simulation. Autoinducer. So we have our quorum sensing uptake, metabolic energy plot here. Fraction of the population by cell type. So it looks like 100% of our cells in this given configuration are I want to say wild type, but I'm unsure. So we can look back. This is what our code looks like right now. We have these global parameters. These are what are going to end up going on our steering window, whether we want mutation. This decay is our metabolic consequence for quorum sensing. Cheating consequence is the defector penalty. Starvation bonus, that is when they're beginning to get low on energy and they decrease the amount that they grow. Quorum sensing inhibitor active, so this is how we turn on and off the effects of the quorum sensing inhibitor without actually removing the field. And these are some other small things that I'll explain. Dilution, but I believe this has to do with some of the different experiments we are running. So whether we want to add new waves of nutrient, the amount of time between this, whether or not cells are randomly killed, and the percentage of cells that are killed. We ended up using this custom defined function called modify value that we found ourselves using a lot of indirect relationships, public good, nutrient, complex nutrient. Modify value made it easy for us to have a ceiling and also just to have change between exponential and linear combinations of these values. We do chemotaxis. So here's our cell initialization, our 3 cell types. And here you can see, so our dictionary entry sensor is whether or not they're quorum sensing. And obviously for unconditional cooperators, they all start as 1. We have our fields. Here's our step function. So here's our dilution information. So you can see it's putting in every so often, it's reintroducing nutrient and complex nutrient. Wild type invasion. So that was from the population graph that Logan showed. So just after 1,000 MCS, we added in a ton of wild type cells. We have our secretors, our uptake. So when we start getting into this big main loop, this is really where we're doing a lot of the grunt work and specifically the changes to metabolic energy. So we take up nutrient, we chemotax, we grow. But yes, here's what I'm talking about. So for each cell, we have our specific decay modifier. We turn on and off quorum sensing for wild type. We make the effect of the quorum sensing inhibitor. We do our starvation bonus. So like I said, we decrease the growth factor if we're starving. And then here we're changing the metabolic energy. And it's pretty similar for the other 2 down here, just with their own decay modifier, their own early quorum sensing, so their own penalties. And then here's our defector. And then once we get down there, yeah, some more plotting, and then mitosis. Like I said, you need a certain metabolic energy to divide, excuse me. And death. So dilution bottleneck, but more importantly, it's how we killed ourselves. And I believe let me look at our PowerPoint before I call it this, there, and done to see if we have anything else. I'll put it up just because we're not. Yes, so thank you guys.
Okay, questions? It would be fast. I mean, I understand the reason to do it on a small lattice. It would be nice to put this up on Big Red and run some big lattices so you could maybe lift some of the number of cell constraints.
Yeah, so I did run some like 1,000 by 1,000 lattice, and I only did that a few times just because I am still working on familiarizing with myself with submitting to the supercomputer, but I did it locally. and let it run for a few days in the background. And a lot of the patterns we were seeing are maintained at the larger ones, but even still, like in 1,000 by 1,000, it's still orders of magnitude off what a real culture is. So it's still limited.
Bacteria. No, I didn't mean to interrupt. Yeah, absolutely. I mean, it's the same problem with cancer. You have to turn your mutation rates way up because you can't simulate 10 to the 7th, 10 to the 8th cells. No simulation mechanism is going to allow you to do that. And so then your one thing that happens is for example, when you simulate cancer, your rate of mutation is fast, so you forget the evolutionary state much more rapidly. And so that's another paradox you could get where I don't think you're going to have that here. But your point about potentially getting apparently misleading trends in small populations is excellent. I'm trying to understand. Can you have an evolution of actually resistance to the quorum sensitive inhibitor, or is that not modeled? You don't have a pump, say an efflux pump, that would allow the cells to.
Yeah, so because the mechanism of import is unknown, I didn't want to model that just because right now at this stage, we're more interested in how it shapes the populations because we all, everyone in the field, based on the data we've seen and the people I've talked to, expected that when I turned that field off or on, for the quorum sensing cells or the wild type cells to no longer dominate the field. And when that happened, we were like, oh crap, I guess we really need to look into this, because even at these really small populations, which people have gotten at with really small culture sizes, we still expected to see that maybe wild type was still the major, but that it should not have dominated in the way that it did. Because, like I said, the expectation was that because recent data has shown that quorum sensing is really heavily linked to the transition between rich and restricted nutrient conditions. We expected that to be detrimental to the advantage conveyed by quorum sensing. And like I said, it could be pleiotropic based on how we're modeling it and the assumptions we're making. But that's why we're using it as preliminary data to justify experiments. And I actually just got an NSF grant to do that based on modeling stuff. So that was pretty cool.
Congratulations. That's fantastic. I guess I was wondering if maybe, I mean, the issue, this specific question about the fact that you see a wild type taking over all the time in the simulation. You begin to wonder whether there has to be some trade-off function that isn't quite what you expect. And so I'm just trying to think of where that could be. Maybe the cost of producing the public good isn't quite as big as you think it is. Maybe the benefits are less diffuse. I mean, there are a couple. I mean, one can think of a variety of possibilities.
Yeah,
probably too many to explore.
Right, and the one thing my committee and I agree that could be an issue here is we created very limited diffusion constants for the public good because they're proteins typically. But the problem is that in bigger cell populations, the liquid is moving, so it's carrying the proteins further. So one thing we've talked about changing is upping that diffusive constant for the public good to see if that maybe changes some things, just because the way the cheater cells are only slightly outlasting the unconditional cooperative cells is not typical. It's not necessarily a red flag because, like I said, the Simpson's paradox could easily explain that away. We're only looking at one given configuration in a very, very nutrient limited cellular population without any actual flow. And microfluidics experiments demonstrate that liquid flow is also important for different quorum sensing experiments. So Nick and I have bounced around ideas for this, but that stuff is kind of outside the scope of this class. It's something we're working together to finish up for a publication down the line. Right?
Sure.
The NanoHub.
I mean, clearly the NanoHub is just for what you're doing, the NanoHub is maybe a distraction, although I think it's not a bad way of sort of clarifying your thinking. Obviously, if you're going to be publishing it, then having an early publication type on NanoHub isn't something you need so much. But I was thinking, I mean, experimentally, could you play with, say, the viscosity of the medium? Could you use an agar or something in the background so that you reduce the effective diffusion constant of the chemicals that are involved?
Yeah, I definitely think that's something we could do. But the primary purpose of the model, as far as like beyond the scope of the class, I think has really been fulfilled in like cluing us into this possibility. Because like I said, no one's really looked at how these things work on small and large population levels in actual native context. So like outside of just media changes, we're also doing these experiments in parallel in an infection model. So generational infections competing defector versus wild type and unconditional cooperator and different population ratios. And this model has been like instrumental in designing those experiments. So that's where it's, I think, really served its purpose as far as my thesis goes. But for like expanding the model for publication, yeah, the auger thing is something one of my committee members brought up. But for like, I work right now, I'm quite honestly, I have so many finals and this project that I'm just focusing on getting that, what we have streamlined. And then, like I said, Nick and I have talked about doing things afterwards that are more outside of what we need to do for this.
Wonderful. Any other comments or questions? People have been very patient. These are all great projects. It's good to see things coming along.